Good afternoon or good morning, depending upon where you're joining us. I'm Brian Cantor, Assistant Research Director at the Duke-Margolis Institute for Health Policy. Welcome to our workshop, Lessons Learned from the Chemistry, Manufacturing, and Controls, or CMC, Development and Readiness Pilot Program, or CDRP, convened by Duke-Margolis and our partners at the U.S. Food and Drug Administration. This workshop comes at an important time, with advancements in biomedical innovation leading to transformative treatments that may come to market sooner through expedited regulatory pathways. These pathways can facilitate earlier availment of treatments to patients and necessitate substantial adjustment to product development timelines. CMC development is a critical component of product development, and it's essential that CMC development keeps pace with clinical development to bring a product to market and get it to patients who need it. CMC issues may hold up the development, submission, and review process for products that may be showing clear clinical benefit. Again, these challenges are pressing for products granted expedited regulatory pathways where all of the components of product development must move in parallel on a compressed timeline. The CDRP program is intended to help sponsors of those products on their CMC development programs and ultimately to ensure patients can receive these therapies, especially when there are few or no treatments available. We'll hear much more from speakers throughout the day on how the program supports those aims, how it has performed through the pilot phase, and what might come next. We are excited to bring together an array of experts from industry and FDA with experience in the CDRP program who can speak to successes, challenges, and opportunities going forward for both the program itself and broader CMC development and readiness. We hope that you in the audience will leave the workshop with a better understanding of the CDRP program, how it has helped support effective expedited CMC development, and how it can best continue to do so in the future. Before we begin, I would like to go over a few considerations for our discussion as well as some housekeeping notes. Next slide. This is the Duke-Margolis Institute statement on our commitment to academic independence. While Duke and the Duke-Margolis Institute do not take partisan positions, all participants here today should feel free to speak their minds on these important topics. You can find more about our policies on the links on this slide. Additionally, we'd like to acknowledge that this meeting is supported by the US FDA as part of our Financial Assistance Award, which is 100% funded by FDA and HHS. You note the relevant language on this slide and that this meeting is not a Federal Advisory Committee meeting. While we are interested in input on this topic, we won't be following any advisory committee procedures or taking any votes. This meeting will be a success if there is an exchange of lots of ideas and open discussion. Next slide. And then a few reminders about our meeting format that will help keep the discussion organized in this virtual setting. First, a reminder that all meeting materials, including the slides from today's meeting, will be available on the Duke-Margolis event website. If you would like to submit a question during the presentations and open discussion portions of the meeting, please type it into the Zoom Q&A function. Please be aware that due to the high volume of meeting participants and interest, we may not be able to get to every question, but we'll do our best to incorporate as many as possible during the discussion. And then a recording of the meeting will be available on the Duke-Margolis event website and YouTube channel after the conclusion of this event. If you have any Zoom issues, please message Luke DeRocher at the address on the slide. We are thrilled that all of you can join us today, and we anticipate an informative and productive discussion. And on the next slide, we'll get an overview of the agenda for today's meeting. We'll start with some opening remarks from FDA outlining the importance of the event, and then we'll follow with three sessions that are a combination of both presentations and panel discussion. The first session will include representatives from the biologic and drug centers at FDA to provide a brief overview of the CDRP program and a few of the key takeaways that have been observed from the pilot to date. The next session, we'll have a chance to hear from several biologics manufacturers that have participated in the pilot program to share their own experiences and thoughts. We'll take a short break, and then our third session will cover a similar topic, but from the drug manufacturer perspective. And then finally, we'll be joined by a few returning panelists in our last session, along with a few new perspectives to reflect on the afternoon's conversations and look forward to next steps. And we'll adjourn for the day shortly before 5 p.m. Eastern Time. Next slide. And with that, I would like to pass it over to Dr. Denise Gavin from the U.S. Food and Drug Administration. Dr. Gavin is the director of the Office of Gene Therapy, CMC, within FDA Center for Biologics Evaluation and Research. Denise will provide opening remarks from the FDA. The floor is all yours, Denise.
Okay, thank you, Brian. Could I please have the, Good afternoon and welcome to the FDA's workshop on accelerating product development. We're excited today to discuss lessons learned from the CMC development and readiness pilot, or CDRP, as Brian mentioned. I'll start off with a brief introduction of why the pilot was developed and why it's important. Next slide, please. This slide just depicts the FDA mission, which is not only to protect public health, but also to advance public health by helping speed innovations that might make medicines more affordable, more effective, and safer, which also includes helping innovators, like the ones we'll discuss today, develop products for serious diseases with unmet medical needs. Next slide, please. To help speed the development of innovative products, Congress and FDA have authorized many programs to expedite the development of promising products for serious and life-threatening diseases. These programs have been quite effective in helping expedite clinical development, but more work is still needed. Next slide, please. The CDRP program is meant to help expedite CMC development. Shown here is a more traditional product lifecycle program where the lifecycle progresses at a more measured pace compared to these rapid methods that we're seeing now. With a traditional clinical study progressing from phase 1 to phase 2 to phase 3 in a more measured manner. And then this allows the sponsor to have sufficient time to adequately characterize the CMC part of the product development. This allows them to gain information about product characterization, to gain understanding of critical process parameters and controls, and to help meet GMPs for licensure. In this scenario, the CMC and the clinical development programs are much more aligned. Next slide, please. Oh, go back one. These programs have okay. In this scenario, for products under accelerated development timeline, sponsors may encounter CMC readiness challenges. Next slide. Yeah, sorry. With rapid clinical development, this can result in compressed timelines for meeting between the early and late phase development, which can present substantial challenges for CMC and successful product development. This is because even though you have an expedited development program, this doesn't change the need to meet regulatory requirements. The sponsors still need to assure the product is safe, pure, and potent, and that it complies with GMP manufacturing. The rapid pace of clinical development can result in a disconnect between CMC and clinical development programs. This is especially true when clinical studies with a large clinical benefit are advancing at a bullet train speed, and CMC lags behind at the choo-choo train speed is progressing much slower. CMC issues can delay pivotal studies as well as approval. We see this for novel products where sponsors may not have as long of a manufacturing history and may have limited product development. But we also can see this when we have experienced sponsors who do not prioritize CMC during early development. It's critical that sponsors align CMC with clinical development to be successful. This essential alignment underscores the need for additional CMC-specific interactions between the sponsors and the FDA, which resulted in the CMC development readiness pilot. Next slide, please. The CDRP program aims to facilitate CMC development of innovative products through enhanced engagement with the FDA. The pilot was initiated in 2023 and should last through 2027. This was a result of PDUFA 7 negotiations where it was clear additional input and interactions between FDA and sponsors was needed to help with CMC development. Many of the sponsors that we have have applied to the program, and some of these sponsors will be presenting later today, along with my FDA colleagues. To be in the program, sponsors need to commit to CMC development at all levels of their organization, not just the CMC review, not just the CMC development team, and to prioritize CMC development early in the product lifecycle. This is for the goal of preventing delays in product approval. Next slide, please. The hope of the program is that increased communication between FDA and sponsors will result in easier access to transformative therapies. In these additional communications, we can discuss strategies to meet regulatory requirements, but we can also discuss options for flexibility in product development, such as concurrent product release, leveraging previous manufacturing knowledge, and various other aspects of process and product control, which you may not understand or may not have enough information yet to see how we might be able to help you with these strategies. Another benefit of the pilot is to facilitate the submission of complete manufacturing of complete marketing applications, which is essential to facilitate review, efficiency, and timeliness. Substantial CMC challenges, have resulted when product development programs, including facilities, were not ready when a sponsor submitted a marketing application. When submissions are incomplete, this can result in a refuse to file decision, can result in multiple information requests, amendments to the application, which can also delay the review and make it much less efficient. This could also result in multiple post-marketing commitments or requirements in order to meet the regulatory standards for safety, purity, and potency. The biggest problem is when substantial deficiencies, they can delay approval, where if you cannot meet the CMC and GMP issues, if these can't be resolved during the review cycle, there will be a complete response letter sent. To avoid delays and increase chances of successful product development, we encourage sponsors to discuss their CMC program with the FDA early and as often as possible. This can be through amendments, it can be through meeting requests, and many other avenues. We also suggest that you consider the CDRP program because it's meant just for this purpose. Next slide, please. In addition to the CDRP program, FDA strives to facilitate product development for rare diseases or unmet medical needs through a number of avenues. Many of these are listed here. We have a number of communication pilots, including as well as the CDRP. The START pilot was initiated last year. We have implemented advanced manufacturing and platform technologies. We are working on international conversions with a number of different programs there. And then we also have stakeholder engagement in workshops like this one, which is very important for CMC development. FDA also implemented the Rare Disease Innovation Hub, which has dedicated staff from all over the agency who tackle these product development issues for rare diseases. In fact, we're planning a workshop on individualized therapies in November, so look for information on that on our FDA website. Next slide, please. So with that promotion of the workshop, let me thank you again for attending this important CMC development workshop. We hope you enjoy our interesting program with interactive discussions throughout the afternoon. With that, I'll turn it over to my FDA colleagues to provide more details and updates on the pilot. Next slide, please. Thank you.
Thank you very much, Denise, for those opening remarks to start us off. And as you said, now we'll hear from some more of your colleagues at FDA in the following presentation to give a little bit more detail on many of the aspects of the CDRP that Denise just previewed for us, as well as, importantly, some lessons learned from FDA's perspective from the pilot phase so far. So sharing responsibility for this presentation from FDA, we will have Ram J. Vatsan, Associate Director in CBER's Office of Gene Therapy, Jennifer Swisher, Supervisory Pharmaceutical Scientist in CDER's Office of Pharmaceutical Quality, and Thomas Oliver, Director of CDER's Division of Product Quality Assessment 1. So I'll ask all three of you to come on camera now, and we can go ahead to the next slide, and you can start us off. Thank you.
Thank you very much. Good afternoon. My name is Jay Watson, and I work in the Office of Gene Therapy in the Office of Therapeutic Products in CBER. I'm joined by my colleagues, Dr. Swisher and Dr. Oliver from CDER. We are all in different product class areas, cell and gene therapies, biotechnology products, small molecules, and will each highlight specific CMC aspects that are frequently discussed during late phase CMC specific interactions with sponsors. Though these product types we will reference may be different, you will notice that there are shared topics that are frequently coming up during accelerated product development, irrespective of the product type. And these are topics that are ideal for discussion during CDRP. Next slide, please. In this talk, we will cover CDRP features, changes that we have introduced into CDRP program based on early sponsor feedback, and discuss specific CMC topics that are frequently discussed during accelerated product development. In the end, we will address some previously submitted questions and hope to have additional time for questions. Next slide, please. Accelerated clinical development frequently do not follow traditional phase 1, 2, 3 development timelines, as Denise just mentioned. Sometimes the phase 2 studies may form the basis for regulatory approval. This reduces time normally available for product development. The aim of CDRP is to ensure that the promising products are not held up from reaching target patient population due to safety reasons arising from incomplete CMC information in the license application. Next slide, please. Cell and gene therapies have demonstrable success in curing diseases that were previously considered terminal diseases. These early successes have resulted in rapid increase in the number of IND applications, as you can see on the left side, the graphs on the left. Successful early clinical studies have resulted in growing numbers of INDs that have RMAT, or Regenerative Medicine Advanced Therapies, which is a CBER-specific designation, and Breakthrough Therapy designations. Based on published information on the timelines for product development, the timeline has come down from 10 to 6 years. To assist companies cope with this rapid pace in clinical development, and in response to industry's proposals, FDA initiated the CDRP program. Next slide, please. The aim of CDRP is to assist sponsors accelerate CMC development to keep up with clinical development. The CMC requirements for ensuring product safety and manufacturing consistency has not been reduced because of rapid clinical development. It is critical to align CMC and clinical development and ensure that the products reach the patient population at the earliest. Next slide, please. The CDRP program has some common features, few CDER-specific features, and some CBER-specific features. Common features include the need for active commercial IND, provides for enhanced communication between sponsors and FDA, explores science and risk-based approaches to accelerate CMC development, and have a commitment to share product development knowledge with others. CDER-specific features are to encourage all sponsors of commercial INDs under accelerated clinical development to apply for the CDRP program, with the final eligibility to be decided by the review teams. Include a facility discussion as part of CDRP, and allocate 1/3 of all allowed applications each year to CDER-regulated products. CBER-specific eligibility criteria include the need for either RMAT or breakthrough designation, and be a CGT or cell and gene therapy or vaccine-related product, and allow up to 2/3 of the maximum allowable number of applications to CBER sponsors. CBER has a dedicated facilities discussion that is independent of CDRP, and this pathway will continue to be utilized for specific discussion on facilities. We encourage you to take advantage of these additional pathways. Next slide, please. FDA published Year 1 FR notice, FRN, at the end of October 22, and the pilot started accepting applications on the 1st of April 23. We received a total of 9 applications in the first year and accepted 3 of them. Year 2 and Year 3 FRNs were published in September 23 and September 24 and have received a total of 10 applications during this period. We have accepted 5 of them. 1 application is presently under review. Year 4 FRN will be published in the next few days, and the program will continue to accept applications through the end of PDUFA 5 period, which is 2027. And all participants are expected to continue to be in the pilot till they submit their license application or decide to withdraw. Based on lessons learned during the pilot and based on the feedback received during the workshop, we plan to publish a strategy document in April 26. The strategy document is expected to describe future plans for accelerating product development and would include information that's gathered during this workshop. Next slide, please. This slide provides a summary of the program status till date. There have been a total of 12 applications to enroll in CDRP received from CDER sponsors, and 5 were accepted. And we have held 4 Type B meetings. 1 applicant has withdrawn for business reasons. A total of 7 applications were received for CBER, and 4 were accepted into the program, and 1 is currently pending, is under review. Based on interim survey of sponsors, we learned that the main concerns from sponsors were related to acceptance review timelines, disclosure letter, and uncertainties regarding available number of spots, and eligibility to enroll in the program. From the FDA perspective, the main reasons for declining to accept sponsors into the program was they are not meeting the minimum eligibility criteria, that is, their progress in the clinical development program. Next slide, please. Based on the results of the interim survey, we made a few programmatic changes, and these were incorporated into Year 3 FR notice. This reduced the acceptance review timeline from 180 days to 90 days, provides for an early involvement of a full complement of review teams to help the sponsors, continuous rolling acceptance of applications without any cutoff period for accepting applications, and allow broader sponsor participation during including both experienced and new sponsors, as well as accept files that may be further along in their clinical development. Next slide, please. In the next few slides, we will try to cover some of the topics that come up during late-stage product development and can be major issues for sponsors who are developing products under accelerated product development timelines. Though many of these topics may appear disjointed, these are all connected, and changes that affect one or more of these topics can affect others. Typically, manufacturing changes are inevitable. Changes such as scale-up, additional manufacturing facilities, manufacturing process changes, etc. occur. Manufacturing changes will typically require comparability studies, additional process validation studies. Validated assays provide higher level of confidence in evaluating comparability. Potency assessment is a frequently addressed topic and an essential component of comparability studies. Depending on the extent of manufacturing changes, additional stability studies may also be required. Readiness of manufacturing facilities are a big stumbling block for many first-time manufacturers. Leachables and extractables for all product contract materials is often overlooked and could delay assessment of product safety and eventual product approvals. Over the next few slides, I and my colleagues will each discuss some of the topics in detail from CBER and CDER's perspective. These will keep in view lessons learned from sponsors' interactions during CDRP and other late-stage studies. Next slide, please. Process validation is an important step in confirming that the manufacturing process is capable of producing the product of desired quality consistently. Though we have a number of guidance documents as listed in the slide that discuss process validations, this is often a topic that requires additional preparation, especially for a first-time manufacturer, and many cell and gene therapy products are manufactured by first-time manufacturers. As you know, there are no shortcuts, and when it comes to establishing product safety. However, to enable sponsors of products that are under accelerated development and through sponsor interactions, we have realized that there could be some regulatory flexibility that would still ensure product safety while accelerating product commercialization. The next slide is a generalization of some of these concepts that are applied on a case-by-case basis and should be discussed during sponsors' interactions with the agency. Next slide, please. Process validation is a GMP requirement, and the process must be validated prior to licensure. Under accelerated product development, there are limited circumstances when there may be some flexibilities. Flexibilities are product, process, and manufacturer specific and cannot be applied universally. This is an important topic that may be discussed during CDRP. Some of the flexibilities can include concurrent release of product that are manufactured as a part of PPQ runs for commercial release. This will depend on a number of additional factors, including tight lot release specifications. This is a topic that should be discussed early, prior to BLA and agreed upon. Process consistency, a part of process validation, should be demonstrated through products that are manufactured at commercial scale. However, under accelerated product development, a scaled down lot that is manufactured according to commercial process may still be used to confirm process consistency. Similar principles may also be applied for the use of reduced number of PPQ lots. These topics should be discussed with the agency prior to the BLA and agreed upon. It may not always be feasible to combine all different flexibilities discussed here in one BLA application. Next slide, please. Another frequently discussed topic is related to comparability and the extent of comparability studies that are required when manufacturing changes are introduced into the manufacturing process. Changes are easier to address when they are introduced earlier in the product life cycle, as opposed to changes that are introduced during or after the completion of licensure studies. We have recently published a guidance that incorporates recommendations on the design and conduct of comparability studies. As explained in this draft guidance, comparability studies help confirm product consistency before and after change. Validated assays, when used to confirm products' critical quality attributes, provide greater confidence in assay results. Product potency is an important component, as I had said earlier, of comparability studies, and retained samples help evaluate the effect of changes in a side-by-side manner and is discussed in the comparability guidance. However, retained sample related guidance may be specific to cell and gene therapy products, while other aspects would apply to generally. Assay validations may not always be complete by the time of comparability studies when product development is accelerated. These studies are performed as part of accelerated product development. The guidance provides generalized recommendations that would apply to a large number of cell and gene therapy products. However, product-specific applications of the recommendations should be discussed during late phase meetings and CDRP meetings. Next slide, please. Product potency is an important critical quality attribute, and assays to measure potency of cell and gene therapy products is an important topic of discussion during late-stage product development and is frequently discussed during CDRP meetings. We have recently published a potency guidance that discusses the great level of flexibility available for products under accelerated development. Potency assays should be part of a comprehensive approach to help ensure that every lot of a product will have the potency necessary to achieve the intended therapeutic effect. It may not always be necessary to have a quantifiable bioassay to evaluate potency when the potency can be assured through a combination of qualifiable analytical methods and bioassays. When a product has a sequence of biological activities, as in the example below, a bioassay designed to measure activity at a later step will often suffice, and additional bioassays for earlier steps may not be necessary. Though it's often common to measure multiple product activities during product development, it may not be necessary to include all different potency assays in a licensed product. The type of potency assay is appropriate for a specific cell and gene therapy product, and setting appropriate acceptance criteria is a topic that's often discussed during CDRP meetings. And with that, I'll turn it over to Dr. Swisher.
Thank you, Jay. Okay. One more time. There we go. Apologies. Thanks, Jay. So speaking from the perspective of a biologics regulator in CDER, the control strategy is really an important piece of kind of any of these considerations. It's the linchpin for any potential risk-based regulatory flexibility that you and the agency might consider together, is really going to be your understanding of your product's mechanism of action and the critical quality attributes that ensure clinical performance of your product. And this is true kind of across the board. This is the foundation for any consideration of regulatory proposals that you might have in this pilot or in any expedited program. It's further built on the product-focused understanding of the manufacturing process, which is gained through many ways, but in these cases, it may be kind of supplemented through prior knowledge or developmental studies. We do understand that for expedited programs, you may not have a lot of manufacturing experience, but you can use prior knowledge and developmental studies to fill in some of these gaps. But it may also be necessary to implement additional analytical methods or additional in-process monitoring, more tests, maybe tighter acceptance criteria to adequately address some of that residual risk due to a lack of data and/or knowledge. Finally, comparability protocols and post-marketing commitments can be tools that allow maturation of the control strategy post-approval, but there still needs to be enough data and information to support approval of a process and a product that ensures continual market supply of a therapeutic with consistent clinical performance and quality throughout its shelf life. I think to answer one of the comments that I saw, one of the questions, the goalposts don't change in terms of how much information or how much assurance is needed in terms of product consistency and the ability to continue manufacturing that product with its expected quality throughout its lifetime. It's just there are different ways to support it. And this program, we look at some of those different ways. Next slide, please. So although not a guidance, CDER does have a map that discusses some of the regulatory flexibilities that CDER and Office of Pharmaceutical Quality will consider in these programs, including a number of approaches that applicants can consider to support a reasonable shelf life for a product with an expedited development timeframe. For instance, based on discussion with the agency, it may be possible to submit an NDA or BLA with less than the typical 6 months if stability updates can be provided during the review cycle. Similarly, supportive data from representative batches of a different size or have other justifiable differences may provide adequate supportive information to help set a reasonable initial shelf life, given a demonstration of adequate comparability to the primary batches. Even justifications from other well-characterized products or the use of predictive stability models, either as directly supportive or the shelf life, directly supportive of the shelf life for small molecules or as part of a comparability assessment can be considered. But all of these approaches, we urge you to have early and sufficiently detailed conversations with the agency to allow us to agree on a plan and give you the time to come up with the data. And I will say that although, the Q1 guidance is in draft and not something that we're kind of operating according to yet, many of those things, many of those concepts are those that are seen in this map that we do consider. Next slide. And in terms of manufacturing readiness, really timely identification of proposed commercial manufacturing sites is incredibly important. And even more so implementation of robust collaborative communication, both in terms of kind of quality agreements with CMOs where applicable, we're testing, labs, but also with the agency. All of those lines of communication are tremendously important. And ultimately, there's kind of no getting around the need to assure that quality systems are designed to achieve sufficient control over the facility and the commercial manufacturing operations there. Changes, trends, deviations, failures need to be adequately documented, evaluated, investigated, controlled, and corrected as applicable. And all of those aspects need to be adequately documented. Facility and equipment controls to prevent contamination, process controls, monitoring, assure low bioburden and sterility as applicable. And again, any flexibility that are considered for proposal should be discussed as early as possible. And I did also want to call people's attention to, there was a meeting facilitated by Duke-Margolis last summer where BLA readiness and kind of BLA quality was discussed, and facility readiness was a big topic. And that's why I would refer you to the slides and kind of the information that's there. There are minutes of those meetings that are really helpful. And then I believe the next slide is Tom Oliver to talk about some of the challenges in small molecules. Thanks. Tom?
Thank you. Thank you, Jenny. My name is Tom Oliver, and I'll touch on the small molecule observations in CDER. In the accelerated programs of breakthrough and fast track, the stability program can often be impacted. And one of the first questions is, does the stability program conform to ICH Q1A? And if the answer is no, then we often see a question in the meeting package in this space about whether or not it's fileable or whether or not there will be sufficient data for a usable expiry. And so good communication is necessary to facilitate timely approval of accelerated program drugs. And what is that? Typically, in a meeting package, we're going to need to see the number of batches, the amount of data at submission, types of information, and the types of data that will be submitted, and then timing of any stability updates. Reviewing of timing constraints is a real issue at the agency. And even though a number of these will come under accelerated review times, maybe 6 months, the clinical division may make a decision to actually act within 4 months. And so we in the quality team may only have 3 months to do a review. And so if someone is planning on submitting an update at month 5, that clearly will be outside the realm in which we'll be able to look at it in that first cycle. And so the discussion of both on the FDA side and on industry side, the communication is very, very important. Sharing drug product formulation and manufacturing knowledge with the agency is very important as well, as we look at information to support an expiry. And known or potential stability failure modes are very, very important to share as well. Oftentimes, companies will be very familiar with a particular type of manufacturing, whether it's wet granulation, and they have an awful lot of knowledge with products in their house. Ultimately, there are very limited outcomes of the review process. There's clearly an AP action. There is an AP action with PMRs and PMCs, and then there's a CR action. And clearly, moving products that move the needle, that affect patients, it's very important to go to an AP action. And clearly, sharing of information at an early point is very, very important. Next slide, please. The next part that we see a lot in the small molecule side is manufacturing considerations. Accelerated development, from what we've seen, creates many gaps in process understanding, analytical method development, and facility readiness. And then we also see at times too where the acceleration of product development may lead to a different site being used for commercial production than maybe what was foreseen months or even years ago. And so things change very quickly. And then including the types of information to support the application is very, very important. So it's important to discuss site and process challenges, what those are, including packaging, the information needed to kind of cover that gap of information. It's important to demonstrate process and product knowledge that you have either on this product or similar products that you may have developed. It's important to ensure facilities are capable and adequately qualified and can generate comparable product. That's very, very important. We clearly look for robustness. Making product at multiple sites can sometimes demonstrate robustness, but it's important to discuss what you have, where you're heading, and how you plan to bridge those. It's important also to highlight risk minimization strategies that you plan to utilize. And it's very important to remember that proactive, transparent discussions with the agency are much more effective than retroactive or reactive steps. And they save an awful lot of review time and interaction with you during the review process. Next slide, please. We received a number of questions early on, and we chose 3 of those to kind of provide here responses to. The first question is, what advice could FDA share with sponsors to help them prepare for a successful CDRP experience? And the answer to this is probably much different than any meeting. It's very important to prepare a comprehensive meeting package with very specific directed questions. Sometimes we get questions that are very broad, and they're not as effective for us to give responses to. It's important to incorporate the recommendations that we do provide and be prepared to discuss any follow-up questions through one of these CMC-focused CDRP meetings, or any alternatives to a recommendation we have can also be discussed as well. The most important way to benefit from CDRP interaction is to invest the time and effort early into getting the product to the market rather than waiting for clinical results to all come in prior to investing in product quality studies. Typically, there's a certain amount of preparation needed to bridge the gaps that you may experience on the quality side, and discussing those with the agency early is very beneficial to have the information that you need to support your application. What are common perceptions about barriers to the CDRP that you would like to address? Some common perceptions include uncertainty of acceptance, and there's a worry about the disclosure agreement. FDA continues to be aware of sponsors' need for confidentiality, and we will not disclose proprietary information. The third question we answer is, how does the involvement of cross-functional review teams in meetings benefit CMC readiness? When an application is submitted for a BLA or NDA, it is reviewed by a quality team, including experience in drug substance, drug product, manufacturing, microbiology, biopharmaceutics, and additional disciplines as needed. The hope is that this reduces the need for many IRs and the length of those IRs. And it also minimizes or eliminates delays in approvals. Thank you very much. And Thomas, I'll kick this back to you.
Yeah, thank you, Tom, and thanks to all three of you for excellent opening presentations here. Great degree of detail that you all were able to share that I know is very helpful to our audience here today. I know we are a couple minutes past the scheduled time for this session. I think we've been getting a lot of great questions in the Q&A from the audience. A lot of those we will be able to bring into our later sessions where we talk more about a lot of these process-related details, drawing from the experience of some sponsors who have participated in the CDRP. I want to take just one moment now to direct a couple of questions maybe to you, Jay, regarding eligibility, since I think that's something that we may not have as much time to touch on later in the meeting. We got a couple of questions about whether FDA can grant acceptance to the CDRP conditionally, perhaps while a sponsor is still awaiting a decision on an advanced or expedited regulatory pathway, as well as some questions about what degree of detail in the development plan you might want to see in an application, and a few other questions along those lines. Can you just briefly share any clarification on eligibility and what degree of preparation you want to see at the point of application?
Yeah, so typically we would expect the sponsors to have an application that includes information on what their state of clinical development is and provide why they would be a good candidate for CDRP and provide some CDRP, their current state of product development, what they have done so far. If this information is present in different sections of the IND that they have previously submitted, they can cross-reference it. So they can cross-reference specific sections so that the reviewer can easily find those sections. And we would like them to summarize their current status of product development so that we have a summary of the information that's been submitted so far. And in addition to that, we would expect that the sponsors have some information on where they're going, how much more, what their plans of future development are. If they are planning to use a contract manufacturer, they should have some idea who the manufacturer is going to be, whether it's going to be a single dedicated product manufacturer or a multi-product manufacturer, so that we can provide appropriate guidance on those topics. In your second part of the question, whether we can have what is the basic requirements for CBER and CDER? The basic requirements are slightly different, but the basic requirement is still the same, that there should be an active clinical program that is at an accelerated rate of development. Now, CBER requires RMAT, our breakthrough designations. RMAT is a CBER specific designation. And so we would expect them to have at least that, because that provides less need for additional support and input from the clinical reviewers who are busy. And we would not require additional time for doing this review. For CDER, anybody can apply so long as they have an accelerated product development. Again, the basic determination is what is different in the two cases. In CBER's case, it's predetermined because they have an RMAT or breakthrough, and for CDER, it's determined at the time of review. Was there another part to this question?
I think that covered it very well. Thank you, Jay, and thank you to Jennifer and Tom as well for your presentations. I know there are more questions in the Q&A there, and some of our colleagues at FDA may be able to answer a few more in writing while we move ahead into the next sessions. And I know several of you will be returning for our later discussions as well. So lots more opportunities to address some of the questions coming in from the audience. Please keep those coming. But for now, I think we can move ahead to the next slide and into our next session. Thank you very much. I'll be moderating again for our next session, which will be focused on CBER-regulated industry experience with the CDRP. We'll be hearing a couple of presentations from industry sponsors who have participated in the pilot already, and then also a presentation from a representative from the Biotechnology Innovation Organization, or BIO, to share a little bit more context on some of their members' thoughts on the CDRP based on some surveys that they've conducted. Following that presentation, we'll have a couple folks from FDA returning to continue the discussion with the three presenters. So I'll introduce our three presenters here. First, we'll have Isabella Palazzolo, who is Senior Director for Regulatory CMC Strategy at Intellia Therapeutics. Then we'll hear from Natalie Ward, who is Director of Global Regulatory Sciences in CMC for Cell Therapy at Bristol Myers Squibb. And then lastly, from Neil Ichiro Leruan, who is Director of Science and Regulatory Affairs at BIO, as I mentioned. So first up, we will have Isabella. Please come on camera for us, and we can go to the next slide and get you started. Thank you.
Wonderful. Thank you. Good morning. Good afternoon, everyone. My name is Isabella Palazzolo. I'm a Senior Director of Reg CMC at Intellia Therapeutics. And I'm also the regulatory CMC lead for NTLA 2002. On behalf of Intellia, I'd like to thank the agency as well as Duke-Margolis for the opportunity to be here today as part of this workshop. I'll be sharing our experience with NTLA 2002 and our journey through the CDRP program. Next slide, please. Over the next 15 minutes, I'll cover three key areas. First, I'll give an introduction about NTLA 2002, and then I'll spend most of my time walking through the detailed steps through our journey through the CDRP. And finally, based on our experience, I'll share some points to consider for the next cycle of CDRP. Next slide, please. NTLA 2002 is an investigational CRISPR-Cas9 gene editing therapy, which is developed as a one-time administration for the treatment of hereditary angioedema, or HAE. NTLA 2002 is a lipid nanoparticle. The drug product is composed of two drug substances, the guide RNA and the mRNA, which are, and is surrounded by lipids, including a novel lipid excipient. The two drug substances provide the key elements of the CRISPR-Cas9 gene editing mechanism. Guide RNA provides target site specificity, and mRNA provides transient expression of Cas9 protein. Both cargo needs to be delivered simultaneously to the cytoplasm, and this is achieved by the non-viral delivery in a lipid nanoparticle. Next slide, please. The mechanism of action of NTLA 2002 relies on an endogenous mechanism for DNA repair called non-homologous end joining. The lipid nanoparticle is delivered through an IV administration and reaches the cytoplasm of the hepatocytes, where the guide RNA and the Cas9 protein complex in a ribonucleotide protein complex. This complex translocates to the nucleus, and there they exert their direct action, which is creation of a double-stranded DNA cut at a specific genomic location. The target gene is KLKB1 gene. Through formation of indels at this gene, we are able to reduce the level of prekallikrein mRNA and ultimately reduce the level of total plasma kallikrein in the blood protein, which is a protein that is a key element in the signaling pathway of HAE. Next slide, please. Now I'll dive a little deeper in our CMC components of NTLA 2002. As mentioned, we have two drug substances, guide RNA and Cas9 mRNA. Those are both synthetically derived oligonucleotides, although the manufacturing process is different as they are different in nature. The guide RNA, being 100 nucleotides in length, is amenable for standard solid phase phosphoramidides oligosynthesis. Cas9 mRNA, which is about 4,400 nucleotides, it's produced through an in vitro transcription using an enzymatic synthesis. In our lipid nanoparticle, we have multiple lipids, some compendial, some non-compendial, and a novel lipid excipients. All of those lipids are chemically derived. And last but not least, our lipid nanoparticle, it's also created by a self-assembly process with controlled ethanol dilutions and combine both RNAs, drug substances, and lipids at the same time. Next slide, please. Now, when the CDRP program was announced, the NTLA 2002 team saw a great opportunity. At that time, we already had orphan drug designation in the US, as well as RMAT designation. And we also had similar recognitions in the other countries. However, the CDRP offered an incredible opportunity to start a dialogue and a collaborations with FDA on a very novel modality. NTLA 2002, as a reminder, remains on track to be the first in vivo gene editing product to reach BLA stage. And at the time of the program being announced, being like actively in phase 2 and about like to start thinking about our phase 3 approach, securing CMC advice was pivotal to maintain our CMC development timeline. Next slide, please. Our application, now I'll walk you through the various steps. So our application came in, our request for application came in June of 2023. However, we were notified around September that there was interest from the FDA to accept our program into the CDRP, but we had to have subsequent clarification on the remit of this exact workshop, the disclosure expectations, which somewhat prolonged the formal acceptance of NTLA 2002 into the program around 3 months. Next slide, please. The formal acceptance arrived in December of 2023. And as part of that letter, the FDA requested an initial meeting to provide an update on CMC development activities and identify topics that we wanted to discuss in the next 2 Type B meetings that we were going to require, as well as timeline for those meetings. The FDA also offered the opportunity to ask additional questions that were not related to the CDRP program per se, but that we may want to ask. Although this meeting came a little bit as a surprise, we really jumped on the opportunity. Next slide, please. The meeting was held in March of 2024. The FDA agreed with most of the topic that we intended to propose for the future Type B meetings and provided recommendation for topics that we had not mentioned that they recommended for our interactions. Those topics included our comparability approach between the ongoing phase 2 study and the phase 3, extractable and leachable, and the level of CMC information to be provided by the non-compendial lipids. And some of those topics you have already heard mentioned by the FDA in earlier slides. We took advantage of these additional topics that sponsor may want to ask and obtain alignment with FDA for our in-use compatibility assessment approach for the drug product that was appropriate for the upcoming phase 3 readiness. And importantly, we obtained clarification about the FDA expectation on potency requirement for drug substance, which I'll expand upon in the next slide. As everyone knows in this audience, potency, like development, qualification, and validation of potency assay is a very time-consuming activity and it requires significant resources and financial investment. Therefore, developing the right potency strategy early is key and specifically for product with rapid development timelines, such as Intellia 2002. As such, we had already, prior to CDRP program application, aligned with FDA on a proposed commercial potency strategy earlier in 2023. However, in February 2024, the OTP town hall for genome editing products provided an update on FDA thoughts about potency requirements. We used the meeting in March to obtain further clarification about the requirement on the drug substance potency testing, specifically for our Intellia 2002 program. As you can imagine, due to the timing of the meeting request and the timing of the OTP town hall, a formal question wasn't included in the initial briefing book or in the initial meeting request, and was only asked during the meeting. Despite the informal nature of the request, the FDA agreed to provide additional clarification on their position expressed at the OTP town hall a few weeks prior to our meeting. The feedback was was instrumental, and we were able to direct the development activities in the right direction in a timely manner. Next slide, please. The first meeting that were planned under CDRP was executed in June 2024. This meeting was utilized to discuss, as recommended by the agency, the phase 1/2 to phase 3 comparability packages for all of our components. We also provided an update on our drug substance potency strategy and obtained an endorsement on the approach. The topic that we wanted to discuss all along was also discussed. We obtained an agreement with impurity control strategy for lipid for our commercial stage. And also we continue to engage FDA in a dialogue around guide impurities control strategy. Next slide, please. I want to mention some key lessons learned and key element of this feedback. The feedback was critical because it was successfully de-risk our phase 3 initiation. The timing of the feedback was critically thought and allowed the sponsor to initiate validation activities for potency methods without waiting for the feedback on our upcoming phase 3 IND amendment. Importantly, the feedback that was received in written response format was so complete and comprehensive that the sponsor decided to cancel the meeting and continue and progress as agreed upon. Next slide, please. The second CDRP meeting was planned and executed in March of 2025. This meeting was focused on endorsement of the PPQ strategy or validation strategy for our drug product, and was also utilized to obtain important guidance for shelf life settings of all of our components. We continue to use this meeting to support the agency understanding of our updated potency strategy for drug substance, and we use that meeting also to continue to receive assurance on our impurity characterization approaches that were earlier discussed. Next slide, please. Again, the timing of this feedback was critically planned, and we were able to initiate, maybe did not save perhaps much time, but definitely provided reassurance on the approach prior to initiation validation activities for drug product, which are a significant financial commitment and could have set us back several times. Next slide, please. Now, looking back to our journey in perspective, we had quite a substantial number of interactions with FDA, and I wanted to highlight that we had also other US Type B meeting happening at the same time, as well as we maintain in our outside of US regulatory interactions path. We also have rolled out a global phase 3 study. So someone asked that looking back, was CDRP worth it? Next slide, please. The answer is absolutely yes. Sure, preparation for 3 CMC only Type B meetings in 1 calendar year requires significant team resources, while the team was busy supporting all the other regulatory and CMC deliverables that needed to occur to maintain the program on time. However, the alignment with FDA in a timely manner for this key CMC topic provided reassurance on approach and definitely helped keeping the pace with the clinical development. Importantly, CMC dedicated meetings allowed more time for clinical and non-clinical discussions at PDUFA milestones meeting that had to occur anyway. The feedback that we received from the agency was complete and comprehensive and allowed us to cancel one of the meetings. But I want to flag that perhaps one of the greatest benefits of the CDRP participation was achieved from the unsolicited feedback, the recommendation that FDA provided for additional topic to discuss, as well as the clarification that was offered for their potency strategy for drug substance. Next slide, please. This is really my last slide, and I would like to leave you all with some considerations for future CDRP program or the evolution of the CDRP. First of all, as you remember, maybe there was a little bit of a delay in us joining the formally being accepted into the program, and maybe a little hesitation up front. So additional clarity on program structure, on this workshop, on the confidentiality agreement and disclosure may increase participation. And I want to say that maybe because we were already a CBER product and already had RMAT designation, perhaps like diversifying, we greatly benefited from 1 hour long CMC dedicated meetings, but diversifying benefits from other designation may entice more participants, and an increase in flexibility on the frequency and scope of those meetings may also be effective, and perhaps considering more frequent but more focused meetings versus broad and twice only. Lastly, I think a 60-day window between the meeting request and the meeting itself is in Intellia timeline, at least like years. And an increased speed in response would really keep up with the rapid development and may increase the benefit. Next slide, please. All of those considerations taken into account, maybe something to consider could be a different meeting format rather than Type B, which has a limited number of pages, different number of topics, different number of questions, and a faster turnaround time. Next slide. With this, I'd like to thank the FDA for the time dedicated to NTLA 2002 during the CMC Development and Readiness Pilot Program, and I want to thank you all for your attention.
Thank you very much, Isabella. A really comprehensive overview of your experience with the CDRP that I'm sure will be helpful to many in our audience. Next up for presentations, we'll hear from Natalie with BMS. Natalie, you can please come on camera now, and here are your slides to get you started.
Thank you. So I'm Natalie Ward. I'm Director of Regulatory CMC here at BMS. I'm very happy to join this workshop today to speak about our experience with the CDRP program to date for arlacabtagene autoleucel. I will caveat this talk that we've had fairly limited experience to date of the program. We have been accepted, but we have not yet had any of our 2 Type B meetings with FDA. But I hope that I can provide some flavor on just why we applied and then the perceived benefits of the program that we see at BMS as well. If you can go to the next slide, please, Thomas. So I'll go through the agenda. I'll provide some context just about the product that we've had accepted onto the CDRP program. I'll very briefly go through the what is the CDRP and then the key decision drivers for why we applied, and also some of the anticipated benefits that we saw as well. I'll briefly go through some of the CMC challenges that we have in the cell therapy space, and then finally just some closing and takeaways. So arlacabtagene autoleucel, or Arlacel for short, is our product which has been accepted onto the CDRP program. So Arlacel is an autologous GPRC5D directed CAR-T cell therapy product currently under investigation for treatment of multiple myeloma. It has a fairly standard manufacturing process for a CAR T. We isolate T cells from autologous leukapheresis from each patient. The cells are genetically modified to express the GPRC5D directed CAR using a lentiviral vector. The cells are expanded ex vivo and then reintroduced back into the patient. And the mechanism of action for the product is that the transduced cells specifically recognize and induce selective toxicity in GPRC5D expressing tumor cells. We have completed phase 1 clinical development, and we are currently in pivotal clinical studies with expedited clinical development timelines to support a marketing application. And the program was granted RMAT designation in December of 2023. So I will whiz through this because I think our FDA colleagues have given us a good background on this, but the FDA pilot was launched in 2023 with the overall objective to facilitate expedited CMC development of products with expedited clinical development timeframes, to increase communication between sponsors and FDA, and then ultimately to provide patients with earlier access to these products. It's really based around kind of early iterative FDA engagement. You get 2 CMC dedicated type B meetings, and then also the opportunity to have follow-up discussion to address any questions that arise from those meetings as well. And it's really focused on de-risking those later phases of development. And also as part of joining the program, as you heard, like FDA have some regulatory flexibility as outlined in the CEDAR map document. Next slide, please, Thomas. So this is just a few of the key decision drivers for why we applied at BMS. Cell and gene therapy products are highly complex, and I think the CMC challenges are very well known in the industry, and these impact both small and large companies alike. Arlocell met the key requirements to be accepted onto the pilot program, having received RMAT designation. And then really what we wanted to do was gain clarity on FDA's expectation for kind of key CMC areas and to minimize the risk of late stage surprises, particularly during BLA review. And what I mean by that is that we wanted to ensure that we had no surprises on our side for what FDA's expectations were for critical kind of CMC development areas. And then also for FDA, we wanted to ensure there was no surprises for them on what our development plans are for the product. So really this aligns with the company's patient-focused strategy, including enabling earlier access to potentially transformative medicines for patients, reducing the risk of developmental delays, and then ultimately just strengthening quality and consistency. So these are a few of the key benefit areas to joining the program that we identified when we were putting the application in. So we get 2 additional type B meetings, and then most valuably opportunities for follow-up as needed, an increased level of agency interaction and collaboration on CMC problems, and then also really to have that kind of transparent collaborative problem solving with FDA to mitigate any knowledge gaps. We also wanted to provide familiarity with the Arlocell program to FDA and then go through key CMC content for a future BLA submission, which we hope would ease the BLA review and then help to potentially de-risk that submission. And of course, we would hope also that that would feed into potential CMC BLA timeline accelerations, really by having that kind of proactive discussion on those key CMC pain points that we might be feeling. So I will go through the application process and timeline. As I said, we're fairly early in our kind of journey as part of the CDRP program. So we go to the timeline below. The Year 2 Federal Register notice for the CDRP went out in September of 2023. We were granted our RMAT designation for Arlocell in December of 23, and we put the application in to join the program in January of 2024. So just to go through what we provided as part of that application, we provided an overview of the product. We gave quite a significant amount of information on the patient benefit and unmet medical needs. We gave an overview of the expedited clinical development timelines that the team had, particularly with regard to where kind of critical CMC activities sat within those timelines. We gave the CMC development history and the current state of development to that point. And then we also proposed the plan and timing for those 2 additional CMC specific type B meetings. And we also proposed topics for those meetings as well. So we applied in January of 2024, as you can see on the top part of the timeline there. We were accepted onto the program 10 months later in October of 24. And as Isabella mentioned for Intellia Therapeutics, we also had some discussion with FDA as well around the public disclosure agreement and just what the nature of that disclosure agreement was. ultimately, we signed that agreement in January of this year. So we are here today at the FDA workshop. We actually have our first CDRP Type B meeting later on this month in September, and we have that second meeting planned pretty close by in early Q1 of 2026. So just to go on to some kind of CMC challenges that we face in cell therapy, these are novel modalities with limited precedent still for even for CAR T products. We have very complex, often very manual manufacturing processes. And particularly for autologous products, they have very unique supply chain challenges. If we have global manufacturing sites that support kind of global access for the product, this all needs to be supported by tech transfer and comparability packages. And just on the comparability side as well, you might have process changes that are planned, including things like scale up or introduction of automation. But you might also have unplanned changes that weren't expected as part of your development program, such as shortage of supply of critical materials or material changes from a supplier that also need to be tackled as well. So potency assay development is also a big issue as well. It's critical that you have quantitative potency assay, which is representative of the product's mechanism of action. And that can be challenging to develop and can take a significant amount of time to do this. Also, with any biological assay, it can be a challenge to validate that assay, and you also have hurdles there. Just on the final point here, expedited clinical development timelines means that often CMC kind of falls behind, and that can cause CMC-driven delays. And I've put this in inverted commas because I don't think any of us in CMC, we don't want to be, and we don't think of ourselves as the sort of slow CMC steam train that that Jay showed before. But we also, it is necessary that we perform the CMC activities that are required for licensure. And when you have expedited clinical development timelines, that often has to happen in a much shorter period of time. And that can create kind of CMC rate limiting issues for submission of a marketing application. And we hope that this program will be something that kind of mitigates that. So just in closing, CMC for cell therapies is often high uncertainty, and we hope that the CDRP program allows that proactive partnership for us with FDA with options for kind of collaborative problem solving. That participation might help to de-risk development and then accelerate access to new medicines. And really at BMS, we thought this was a good strategic decision, both for science and also for enabling earlier access of these transformative medicines to patients. Just in closing, I'd like to acknowledge the Cell development team because they do an incredible amount of work on this program, and then also FDA for their continued support as well.
Thank you so much, Natalie. Excellent presentation. And now we can move ahead to our final presentation for the session. Before we get into the discussion portion, we will have Neil from Bio presenting, as I mentioned earlier. Neil, please go ahead and come on camera, and you can start when you're ready.
Thank you, Thomas. My name is Neil Larwan. I'm a Director of Science and Regulatory Affairs here at Bio. And I also have the pleasure of being the policy lead staff for Bio's manufacturing committee. And so I want to first thank FDA and Duke-Margolis for the opportunity to speak on behalf of Bio, as well as our biotechnology members. Next slide, please. Before we proceed, of course, I just wanted to share who we are quickly. We are the premier biotechnology advocacy organization that represents biotech companies, industry leaders, and state biotech associations in the US as well as around the globe. And we have our membership ranges from the small biotech startups and to the world's largest pharmaceutical companies, all united in the same goal of developing medical and scientific breakthroughs that improve patients' lives. You may also know that Bio organizes the Bio International Convention, which we had earlier this summer, and as well as a series of annual conferences that drive partnership, investment, and progress within the biotech sector. Next slide, please. And so we'd want to first start off by saying that we appreciate and support FDA's efforts in aligning CMC readiness with expedited pathways through this year, the CDRP program. And this very much aligns with our member companies' values in terms of how they see early engagement with FDA. And they also appreciate and see the need for regulatory flexibility and novel modalities, especially when it comes to biologics and cell gene therapy products. for the sake of reaching patients faster. And so overall, Bio sees the value of the CDRP program, sees it as a tool and potential tool in accelerating the products forward. Next slide, please. And so how did we get here? First, Bio has always had an interest in assessing the current status and progress of all FDA pilots and programs. And so this includes the CDRP program when it was announced in 2023. And so in 2024, Bio and Pharma conducted a joint survey that assesses the industry's awareness of the pilot, as well as their intentions in terms of participating, whether they intended to or not to. And then we did a follow-up survey earlier this year that spoke more specifically to the actual participation in terms of those that actually went through the pilot, as well as any other barriers that companies face, whether when they applied or did not choose to apply. And so overall, with both surveys, we provided real-time insights overall in terms of how the industry saw the CDRP pilot program in terms of awareness, use, and its barriers. Next slide, please. And so here, I just wanted to share quickly the snapshot of the survey results and findings. And so in the initial survey that we did, we received and heard that about 79% of the respondents mentioned that they were aware of the CDRP program. And of that 79%, 57% shared that they were considering applying and had interest in participating in the pilot. This shows that there was reuptake in terms of awareness and consideration, and that makes a lot of sense given that this was a PDUFA 7 commitment. And then so in the follow-up survey, we sort of discussed what happens in terms of the applications throughout the process. In our follow-up survey that we started earlier this year, after about 2 years of accepting applications, we received 3 instances where members shared that they applied. So 3 companies of our shared that they applied. And of those 3, those were intended to be a biologic product and to be reviewed by CBER. And they shared that 2 of the 3 were accepted to the pilot. And so here again, seeing that there was a lot of interest from the initial survey, but then sort of seeing that there were only so few that actually applied. On the other side of the survey, we asked sort of what those barriers could be, and that's sort of on the next slide. And so we had questions asking participants on the survey why or why not they would consider not applying. And these are the reasons that they cited reasons for. One, of course, is that seeing that it would be an additional burden to apply, the idea that it would be too burdensome for them to apply for another program, given the fact that it's an additional to their RMAT or breakthrough designation. They felt that it would be an additional thing, given the fact for the other reasons listed here, is that they were unsure of the benefits compared to what the program already provides. And so given the fact that they were unsure what other additional benefits to those those additional meetings would be, companies were unsure whether they want to provide more time in applying. Another reason that they felt, of course, is the eligibility criteria. Again, for our CBER biologic product, it is a full requirement of RMAT designation or breakthrough therapy designation. Meanwhile, in the CDER product, it's a little bit, more flexibility is included there. And then, as well mentioned, as Jay mentioned earlier, the criteria on the earlier Federal Register noted that it's preferable to smaller companies or companies with less CMC manufacturing experience. We provided this feedback early on to FDA, and very much happy to see in the most updated FRN that criteria has been broadened. So very much appreciate FDA in terms of expanding that criteria. And last but not least, as Isabella and Natalie mentioned earlier, there were some concerns on the confidentiality and disclosure terms. And so this prevented companies and sponsors from considering, given those were unclear. And so with that, if we can go to the next slide, we were able to sort of come up with 3 main recommendations in terms of how the pilot can be refined. One, of course, again, is to clarify those confidentiality guardrails and disclosure agreements, clarify what those agreements are expected and to what extent of those disclosure agreements can be negotiated with the sponsor. Having those clearer will lessen uncertainty, and then sponsors will more likely participate and apply. Second recommendation is showcasing and highlighting CDRP's unique value. Again, given there are breakthrough and RMAT designation, what are the additional benefits? Further distinguish what the benefits could be with those additional 2 meetings, which, again, Natalie and Isabel have shared their perspectives, and so further highlight that and include publicize that further. And then last but not least is to share aggregate outcomes and then further integrate learnings into review practices. Again, very much grateful and appreciative of the companies that are sharing their experiences today. Of course, the integral part would then be to take out those best learned practice lessons learned and then to fully implement them into practice. And then I believe next slide is the last one. And so with that, I like to conclude that Bio fully supports and appreciates FDA's efforts and believes in the CDRP's goals to accelerate product development and enable patient access. The survey that we conducted showed that there is a lot of interest within the CDRP program, and that, however, there are barriers that prevent enabling of the program to further be efficient. And so, and then Bio looks forward to continue collaborating with FDA and refining the pilot and adoption future lessons learned into practices. And I believe that's all I have. So thank you very much.
Thank you very much, Neil, and glad that you ended there on reiterating some of the important goals of this program, and appreciate you raising some of the challenges or perhaps points of hesitation that some folks in industry are feeling as well. So with that, I think we will proceed into our moderated discussion here. Thank you for pulling up that slide. All 3 of our presenters there can please come back on camera, and they'll all be joining us for some further discussion. We'll also be joined by a couple of participants from FDA, as you can see there. We welcome you to join us now. Those will be Andrew Burns, the Director of the Division of Gene Therapy 1 at CBER's Office of Gene Therapy CMC, and Kimberly Schultz, who is Director of the Division of Gene Therapy 2 at CBER's Office of Gene Therapy CMC. And so the both of you, Andrew and Kimberly, please come on and join us now. And we can take this slide down as we jump into discussion here so we can see everyone's faces. And I wanted to start off building on some of the points that Neil raised there, actually, around some of the challenges or points of hesitation that some sponsors who could be potential participants might be considering, and maybe try and draw out some lessons learned from the experiences of the 2 presenters who have participated or are entering the CDRP program. So on that point around kind of differentiating the benefits of the CDRP as opposed to some other expedited regulatory pathways that maybe already offer some additional engagement with FDA. I think that's a point that Neil raised, and we've seen some questions about that as well. I think that would be a really useful one to elaborate on for our audience here. Natalie, maybe can I pass that to you and ask you to talk a little bit more about the decision from BMS's perspective to apply? I know you highlighted some of the benefits you hope to achieve. Can you talk, when you were working through that decision internally, how you viewed the potential benefits of the CDRP as different from some of the other pathways available?
Yeah, absolutely. That's a great question. I think in terms of just getting specific CMC advice, there are mechanisms that you can go. We have RMAT designation and we can go for type B meetings. I think having that focused discussion, and also for us, and it's really encouraging to see Isabella talk about Intellia's kind of informal and getting kind of feedback on things that you didn't ask about as well. And I think the perceived benefits for us is to have that kind of ongoing transparent discussion with FDA, so that if we do have issues that come up in late stage development that we didn't expect, like the unplanned changes that I kind of mentioned, that we can get kind of timely feedback on this.
Thank you very much. Yeah, that's really helpful, and agree. It sounds like kind of de-risking this process and having a little bit more certainty at earlier stages sounds like a major benefit. And I'm glad you raised the point about informal feedback as well. I think that's an important one that we've gotten a lot of audience interest in too. Isabella, can you maybe just remind us, I know you touched on this in your presentation, that some of the greatest benefit was from those unsolicited feedback or questions that you heard from FDA. Can you remind us, FDA kind of raised some points that they kind of directed your attention to that they felt were important in the CMC development process for you. Is that correct?
Yes, I think during the initial meeting, we provided a list of topics that we wanted to discuss and we thought they were important to keep our CMC development on time. And we were provided a list of additional recommendation of things to consider, which we have been working actively upon in the past 2 years. So that definitely was extremely helpful. They may not have been in our forefront and they have been paid attention to instead.
Absolutely. And maybe Andrew and Kim can follow up on these points as well around how FDA can provide some additional feedback on CMC development, both through the Type B meetings and other kind of informal avenues. Can you talk from FDA's perspective about how you determine appropriate opportunities to provide some additional feedback?
Yeah, I can start and then Kim can chime in, but I just want to echo what we've heard today. It's great to hear the feedback on what we've done well and what we can do better on. One of the benefits of the CDRP program from our perspective is that we get to see a top to bottom view of the entire program. And that allows us to give that proactive advice. We get into the mindset of, this is going to eventually be a license. What should we let the sponsor know about now so that they can work on it and we don't discover it later during the license application when there's no time to fix these issues. So that's the really unique and valuable thing about this program for us. But there's also a lot of other opportunities to get CMC advice, more focused advice. Type D meetings are an opportunity, for example, to have a very focused discussion about one discipline. So that's something we see quite often, for example, for a potency assay discussion or a comparability discussion. Those are open to any program, regardless of designation, and we're happy to have those discussions. And we also have opportunity for written feedback after meetings if there's additional follow-up questions that need to be answered. Kim, would you like to chime in?
Yeah, sure. I agree with everything Andrew said. I just want to add on to where he talked about the importance of the CDRP program is really that we get to see an overview of where you are and where you're going. And so our review teams also look at that information and try to find out what additional questions we might have or what additional information we might need in order to get better clarity. And then that leads to our review teams actually recommending other ways to interact with us, either through submitting an amendment or a Type D meeting, or if there's facility concerns that we have, recommending like a Type C facility meeting, something like that. So it's really an interactive process for what those other interactions might look like as well.
Appreciate that. Thank you. And now to dig in. Oh,
Maybe,
please, Isabella.
maybe I want to add, echo to these points that the FDA just raised. I want to say those briefing books were significantly different than a normal briefing book for a Type B meeting, and somewhat made us to some degrees uncomfortable because running through your CMC development milestones, what you're doing, what you may be doing, what you're planning to do that may not come true. That was definitely, we had a significant, maybe inverse proportion. It was a lot of background and very maybe targeted company positions, where for a normal briefing book, we would have a general background and then a very elaborated company position to explain the question. I think if anything, we took really an opposite approach. Our briefing book were extremely more like a story rather than a lot of background information for sure. And they maybe have helped the feedback that we received.
That's a really useful point. Thank you. And maybe we can follow up on some of the preparation work that you were just touching on that went into those Type B meetings. And I know Neil had mentioned that concerns about administrative burden associated with the program are one thing that some sponsors have mentioned. So can you talk a little bit more about how you manage the additional work of preparing for these Type B meetings, and then kind of looking back on it, how that all balances out with the benefits that you received from additional feedback there?
That's for me, right?
Yes.
Yes. I think I kind of like answered the question already. Like the preparation for these meetings were pretty intense. I have to say, like in one of the like it was a balance between trying to get a complete and comprehensive package. At the same time, like we really wanted to have the response on time to the question, because we were moving at like a very fast pace. So like how far we take the story to add to ask a question. So ideally, we would have loved to have some more data to put in our briefing box before asking the question to help the FDA. And like we tried to the best of our capacity to put a complete package together, but definitely that was a struggle. And I think like upfront, like a clear planning of what question do we want to ask and at what time do we want to ask them really helped us like drive into the preparation of those briefing books. For sure, like having a clear map of 2 years of activities, what's going to happen in the next 2 years, was probably like the best time spent in preparing for those meetings.
Mm-hmm. That's helpful. Thank you. And Natalie, can you maybe elaborate a little more on how that's looking from your perspective as you look ahead to planning out those milestones and thinking about where FDA feedback through the CDRP will be most impactful? I know you mentioned comparability maybe is one important point that you'll look to hear from FDA on. Any thoughts in that direction?
Sure. So, I mean, we took a long time to get accepted onto the program. And so I think our development is going at speed. As Isabella said, there's no stopping it. So I think in terms of when the meetings are held, I mean, it's really important with any health authority meeting that you have that you get that feedback in a timely way so that you can feed in those recommendations. Just on the comparability front, comparability studies for complex products is always challenging, and we haven't had any feedback specifically through CDRP yet regarding comparability, but we would always look to gain scientific advice before we complete those studies so that we get agreement on things like number of batches, statistical analysis being used, setting comparability acceptance criteria, and then to feed those recommendations back into the study. What you don't want to do and what will cause delays is if you don't do what FDA have recommended that you do and feed that in so that you end up doing it twice, which is something you do want to avoid.
Mm-hmm. Yeah, absolutely. Andrew and Kim, anything you would want to add on FDA's advice for best practices on comparability? Obviously, avoiding having to do it twice, I think, is great advice there. But anything you want to add on best practices for comparability?
Sure. Comparability is a huge topic, and these studies are often very difficult. These are complex, novel products, maybe not well understood in some cases. There's small numbers of lots, and clinical development is moving very fast. Sometimes manufacturing needs to be moved to another facility or another CDMO. So this comes up a lot, and it takes up a lot of our review time, and we put a lot of effort into it as well. I think sometimes it requires multiple discussions, like the first iteration of a comparability protocol is too vague or is going in the wrong direction. We provide some pointers, so sometimes that can require multiple meetings. We have a draft guidance on comparability, and we encourage people to read that carefully. Also, it's got advice on having discussions with the FDA, so we're happy to have those discussions. This is a huge part of our responsibility here, is to guide sponsors in the right direction in coming up with comparability studies that can be acceptable. And I know this is quite complicated, especially for cell-based therapies, so I'd like to ask Kim to chime in.
Yeah, my division focuses on cell-based gene therapies, which the variability of the starting material, particularly when you're talking about autologous lots, can add additional complications on top of the already complicated topic for comparability study design. And so we really appreciate when sponsors come in and talk to us and give us their protocol ahead of time. And so we can give tailored advice based both on the product design, the changes that are being made, and then also where you are in your development timeline on what type of comparability study you would need. And so across the board, we would recommend putting those changes in as early as possible. And then we can work with you in possibly reducing the expectations for that comparability study. In particular, when you're talking about cell-based gene therapies, when you're moving later into clinical development, having that data is more critical. And so we would push towards a split apheresis type of design and a toast, and that's going to add to the complication of the protocol that you're submitting.
Thank you both for additional context on that. And I'd like to bring Neil back into the conversation here. We've touched on comparability and a couple of other particular parts of the CMC development process that can be challenging. Any others that we haven't touched on that you've found are top of mind for any bio members, either through surveys thinking about what the CDRP could be most helpful with, or just other important CMC challenges that you think would be worth discussing here?
Not in particular, aside from I think the main issues were discussed was really looking at those benefits in terms of how the differences of that. So I think we discussed those pretty well.
Appreciate that. Thank you. Then maybe zooming out to a little bit of an international perspective. I know, Isabella, you had mentioned in your presentation that Intellia was balancing these interactions, additional interactions with FDA through the CDRP, as well as interactions with EMA as you were seeking access to multiple markets for this product. Maybe actually, Neil, let me give you another chance to weigh in here as on how industry generally is kind of navigating that global regulatory environment, particularly in cases where development is expedited, and then would love to hear more from really all of the rest of the panel on your perspective on the same points.
Yeah, for sure. Thanks, Thomas. I think given the fact that, when you think about sponsors going globally, you're going through multiple regulatory authorities, not just FDA, they're going to have different expectations. And especially for, cell gene therapy, each of them will have their own variabilities there as well. So having to maintain what are the regulatory, differences between each of regulatory differences, but then to also meet the needs for each of their biological requirements. That's something that, sponsors will have to manage. And so that can be a lot of work and a lot more costly, which sort of leads to the need for harmonization. And that's something I can share in terms of activities within ICH. That is something that ICH actually will be eventually looking into, which is the comparability of manufacturing changes for ATMP products. And so that was actually Bio's proposal that we proposed last year that was accepted to ICH and will be expected to start later this year. And that is, again, looking at that problem of comparability and looking at how the various regulatory health authorities look at that issue. And so that sort of shows a need for such activities in terms of harmonization. But in addition to that, there's also the ICMAR pilots in terms of collaborative assessments with multiple region health authorities. And so having those activities be streamlined and continue to be active on, that's sort of where I think there's a potential in terms of how to make it much more accessible for sponsors to look at that issue.
Yeah, absolutely agree on the importance of that priority and some of the promising work recently from ICMAR and ICH and others. I know we're maybe a little bit beyond the scope of the CDRP there, but obviously an important consideration for sponsors in their development. Back to Isabella and Natalie, any thoughts on how you balanced these expediting the process and your increased engagement with FDA and how that went in parallel with interactions with other international regulators?
Yeah, I think it's I just maybe like a very simple answer. It's a lot of work. Like obviously as an industry, we want to develop products that are acceptable globally. So like for us, acceptance of our package globally, it's pivotal. It's really important. So we did continue to engage with EMA as well as UK as major markets. Where we were running also our phase 3 study. So we continue to And I just want to say, for us at least, we did find some inconsistencies. We worked through them, and that created like another like maybe tangential effort that needed to be supported. But like probably differently, there may be some cell-based therapies. I think like the agency's expectations have remained pretty much aligned. Like I would say like 80% like of the content would be aligned. Obviously, that 20% could be very impactful, but like an harmonization is definitely welcome at any point in time, reducing those tangential discussions that need to be spiral to maintain the program marching towards a common goal. I think that definitely will be welcome. But maybe compared to other maybe I don't know, Natalie has something else differently to say, but like for our product type with like oligonucleotides type of drug substances and a lipid nanoparticle that is not a viral vector drug product, I think like we have been roughly aligned on our CMC package.
Yeah, I will echo everything that Isabella said. I mean, harmonization is always very welcome in this field, particularly where you've got very novel products that are new as well, where you've got kind of divergence in regulations and guidance. On our side, I will say, you know, it is important to us to go for a scientific advice in other markets where we would be expecting to submit a marketing application as well. And just on the burden piece, I mean, it is a lot of burden. It takes a lot of time to put these meeting packages together. It takes a lot of time to do the prep, and it's a balance really on, you know, you can't have your team constantly going on scientific advice and not being able to do the work to support development of the program. So it's about having that balance and just making sure that you're going the sufficient number of times and asking the right questions so that you get the right feedback that you need.
Mm-hmm. Mm-hmm. Great points. Thank you both. And Kim, Andrew, anything you would want to add on the international perspective? I know FDA does have frequent interactions with some international regulators, and it sounded like from others' comments, for the most part, there is alignment. I guess in some cases, inevitable slight divergences, but any thoughts on how FDA works on aligning regulations with others?
Yeah, we do. So we participate in, ICH, of course, and we're very excited about the comparability guidance development there. And we also have mechanisms for talking to other regulatory agencies behind the scenes. So we do ask for advice and try to align on things. The only and we also have a formal mechanism with EMA, the Parallel Scientific Advice mechanism, which does not get used very often because I think because it lengthens timelines, but we have found that to be helpful for aligning on things. I wanted to mention, if you are seeing discrepant advice from regulatory agencies, we aren't always aware of it. So if there's something that another regulatory agency tells you that doesn't align with what FDA is saying, come talk to us about it. Let us know what that advice is. We are not always aware of the advice you're getting from other agencies, and in some cases we can do something about that.
Really useful advice. Kim, anything to add?
Nope. I think Andrew did a great job answering that.
Excellent. Then before we wrap up for the session, just a few minutes left here, I want to touch on one more topic that has come up a bit tangentially through some of the presentations here and was mentioned more explicitly in some of the opening FDA remarks, which is the importance of coordinating across manufacturing sites through the CMC development process. And I believe one of those opening FDA slides cited a meeting that me and some of my colleagues worked on last year at Duke-Margolis around similar CMC issues, and coordination with CMOs in particular came up frequently there. Neo, maybe we can go back to you to start on this one, since I know this is really something top of mind across industry, the importance of coordination across manufacturing sites, whether those are in-house or contracted. Can you talk about how some of your members might be thinking about that process and any best practices perhaps that you can share for ensuring coordination there, ensuring site readiness, especially in cases of expedited development?
Yeah, for sure can try my best. I think given the fact that our members are very much, there's a wide variety of our membership. We have some that have their own manufacturer, they have multiple sites, and we have some that, will depend on CDMOs or, to do their own sites. And so I think it's very much, first of all, key for sure to be, engage in those discussions and communications in terms of awareness, in terms of what is happening with the regulatory side, as well as on the manufacturing side, ensuring that those are very much so aligned. I think that those are things that we've been hearing from our members in terms of the issues that they have to deal with, as well as the ones that we've already discussed. But I'm sure probably our industry speakers here could probably have a better light in that.
Yeah, please go ahead. I think Natalie, your slides touched on this, so maybe looking ahead how you're planning for ensuring coordination and strong quality control across sites.
Yeah, absolutely. I mean, if you have global reach for your product, then you have multiple manufacturing sites across the world, then yeah, ensuring that you have the tech transfer and comparability packages to support that is really critical. And autologous products particularly, I think, feel this pain in terms of ensuring then that, you know, you are making comparable drug products in every manufacturing site that you have, and that, you know, you can pull that clinical data to support marketing application, and that, you know, you ensure that you have consistent product in routine commercial production as well. So yeah, it's an ongoing challenge, I think. And yeah, just one that I think is going to probably be more well developed, I guess, as the industry grows. So
Absolutely. Kim, I saw you came off mute there.
Yeah, I'd just like to add to that. At the FDA, we have quite a bit of experience, particularly in cell-based gene therapy, working with sponsors who have multiple manufacturing sites and different strategies on how they're controlling it across the sites. And so we're happy to talk to sponsors before, you know, they start their manufacturing to look at comparability protocols and to look at how they're managing that oversight in order to ensure that it's comparable when they launch those sites, and that they're doing proper trending throughout to make sure that they're maintaining that comparability, so that they're really providing a quality drug product across their sites to all their patients.
Appreciate that. All right, we are just about at time for the session. Any closing thoughts from anyone? Anything that we have not hit on yet that you would like to raise before we go to our mid-afternoon break? All right, then I will thank all of our panelists and presenters for this session for sharing your expertise and insights with us. Really productive discussion, I think. We will take a break now for... Hello, everyone, and welcome back. We will resume here momentarily with our next session, which will be focused on CDER-regulated industry experience with the CDRP. We heard just a few minutes ago from folks who participated in this program under the CBER-regulated side, and now we will hear from a couple of participants, sponsors with CDER-regulated products who have participated in the pilot stage of this program. I'll have the pleasure of moderating this session as well, and we will have a couple of new presenters for you, industry sponsors who participated in the pilot. Those will be Claire McCarthy, who is the Senior Director for Regulatory Affairs, CMC at Bicycle Therapeutics, and Alyssa Carter, who is the Senior Director for Regulatory Affairs, CMC at Rayze Bio. And then joining us for the following panel discussion, we will have a couple of FDA participants there as well, who I will introduce when we get to the moderated discussion portion. But for now, I will go ahead and let Claire get us started. Claire, can you please come on camera for us? And we can go to the next slide, and you can go from there.
Hi, thank you very much. So thank you. I'm delighted to have the opportunity to present today. To introduce myself, I am Claire McCarthy. I lead regulatory CMC at Bicycle Therapeutics. Some of my presentation covers similar themes to those already, so I won't labor those points, but I hope I can provide a few additional perspectives today. I'd like to move to the next slide. I'll start with a little bit of background about Bicycle and then what our Bicycle platform is. So we are developing bicycle molecules, which is a novel synthetic peptide modality, which is based on Nobel Prize-winning science that span out of the University of Cambridge. The majority of our development is based on oncology indications, and our lead asset, selinexor pevedotin, that's part of the CDRP program, is being investigated in multiple cancer indications. We also have a pipeline of radiopharmaceuticals designed to address novel cancer treatments. And our company is based in Cambridge in the UK and Cambridge, Massachusetts. Next slide, please. So bicycle molecules are formed by constraining short linear peptides into stabilized bicyclic structure using a central chemical scaffold. And this constraint has the potential for very high affinity and selectivity to a potential target. In addition, it has a large surface area that's presented by the molecule, and it hopes that these bicycle molecules will allow targets that have been unsuccessful in the past with small molecules can be targeted by our bicycle molecules. Also the approach of the bicycle molecule is it will mimic protein-protein interactions. The other advantage of bicycle molecules is that they can be readily conjugated to other payloads or lead to other bicycle molecules without losing their properties. And we really, our aspiration, these molecules can be thought of combining the pharmacological properties normally associated with biologics, but with the pharmacokinetic and synthetic advantages of small molecules. Next slide, please. So bicycle molecules are developed using our phage display platform, which is a proprietary screening platform, and it uses synthetic biology to display a huge number of initially linear peptides on the surface of an engineered bacteriophage. Those are then constrained with scaffold to form the bicycle molecular structure on the surface of the phage particle. We can then make various adjustments to the bicycle molecule in terms of the number of amino acids, the symmetry of the loops, the choice of scaffold to increase the diversity, increase the number of options we have so that we can find a bicycle molecule that is very selective for a desired target. Once we've identified a bicycle that binds to our target, the monomers are then optimized. We can make them on peptide synthesizers and use some non-natural amino acids as to replace some of the amino acids to improve affinity to the target or add some desirable properties to the molecule. And then the final stage, really, when we have our desired bicycle molecule, is to put them together as a therapeutic candidate. And we can do that by adding on payloads or indeed other bicycles. So if you want to move to the next slide, the flexibility of our platform really allows us to develop a lot of different drugs for different targets quite rapidly. We have and currently got 3 types of bicycles in development. Bicycle drug conjugates, where the bicycle is conjugated to a cleavable linker and a cytotoxic payload, and those are used to deliver the drug to directly to the tumor using a specifically targeted bicycle. We have bicycle radioconjugates, which are essentially analogous to the BDCs in that they deliver DNA damaging radiation directly to a tumor using a targeted bicycle. And finally, we have bicycle molecules in the form of multimers. So the bicycle TCAs, tumor-targeted immune cell activators, where they designed where the immune cell agonist is delivered directly to the tumor cell using a targeted bicycle. So those are where you have 2 or more bicycle molecules combined together. So quite a diversity of different types of drugs that can be developed from the bicycle platform. Next slide, please. Zelenectide pevedotin targets Nectin-4, and it's been which has been shown to be overexpressed in tumor cells and is believed to play a role in tumor cell growth and proliferation. And the zelenectide pevedotin is a BDC. It comprises a Nectin-4 targeting bicycle, a cleavable linker, and an MAE-based toxin payload. So it's currently been evaluated in several company-sponsored clinical trials. We have Duravelo 1, where it's studying zelenectide in several Nectin-4-driven solid tumors, while a phase 2/3 Duravelo 2 study is for urothelial cancer. And then we also have Duravelo 3 and 4, which are studying zelenectide in Nectin-4 amplified breast and lung cancers, respectively. So one of the key criteria, obviously, for entering the CDRP program is to have an expedited designation. And at the time of the application, we had a fast track designation in urothelial cancer for zelenectide. And now we have since then received fast track designation in both breast and lung cancer. I'll move to the next slide. The fast track designation has made us eligible for CDRP, but why did we particularly want to join CDRP? And while bicycles are a hugely promising platform for drug development, the nature of the molecules means they have some specific CMC development challenges, and specifically the BDC does as well. So you can see here it has, for a small molecule, a relatively large molecular weight. It has the constrained bicyclic peptide, and it also has got the MMAE payload. So it is reasonably complex molecule, and it is, there's a number of aspects that add to the complexity from a CMC perspective. Next slide, please. Taking a step back, I guess, as well, I think, and several people have covered this today, we know that there is a known issue, and that's why CDRP was started, where the CMC is not keeping pace necessarily with advances in clinical development. And we saw that even recently with the last tranche of complete response letters published by the FDA, where I think in July the reporting was 74%, so nearly 3/4 of applications, it was quality or manufacturing issues that were driving that. Clearly, we don't want to hold up access to innovative medicines due to CMC challenges. It's critical for the sponsor company that we can bring these molecules to market, and for FDA, but most importantly, it's critical for patients that we're not holding up promising clinical advances due to CMC issues. Of course, the other thing we need to bear in mind is, the requirement of a quality product at the time of approval, and that requirement doesn't change no matter whether there is an accelerated approval. So we've got to consider that as well. Next slide. So as soon as CDRP was announced, it was clear that it was something that Bicycle would be really keen to take advantage of. We had planned expedited clinical development of zelenectide. We were keen to de-risk our CMC development. And from my perspective, it's important to try and ensure that CMC isn't going to be the reason why we get delays to approval. In addition, as a smaller sponsor, a UK company, it was really important to us to be able to signal externally that we've taken steps to de-risk our CMC program, that we'd a path to success with the FDA in CMC. So this is something that was hugely important to us, both from the opportunity for the additional interactions and advice from the FDA, but also as part of our, perception of our product and our development as well. If you want to move to the next slide, please. I can talk a bit through our experiences with CDRP, and I think it looks quite similar from here, from what people have already talked about today. I think we applied as soon as the CDRP program opened, and that was because we received our Fast Track designation in urothelial cancer in December 2022. And as soon as we received that, we gathered a team together for our CMC team to try and put together the CMC development plan in line with the guidance that was provided for the program. So this is a reasonably big piece of work for us to put together our CMC development plan. We wanted to ensure we covered all the sections that were in there and provided all the required information. And actually it was useful for us in several ways as well, because it allowed us to really think about our CMC development in a structured way, what our plans were, and helped us draw out some of the gaps that we could see that we may require further agency input on. So once we submitted, we were accepted onto the CDRP. I think it was just over the 180 days following the completion of the disclosure agreement. That acceptance letter didn't include any specific feedback on our CMC development plan, which was for us, I think, a lot of work had gone into that. There was a lot of rich information, so perhaps there's an opportunity there for that. It did, however, include a request for us to have a first CMC-focused meeting under the pilot to identify issues for discussion in the two additional type Bs under the pilot. So we updated our CMC development plan, and we submitted a meeting request. However, prior to the granting of the meeting, we received feedback from the FDA that actually we should update our briefing book to include specific questions on CMC topics, and that this meeting would be actually considered our first meeting under the pilot, our first of two type B meetings. So I think a slightly different experience from perhaps some other applicants under CDRP. We used the CMC gaps or the issues that we'd identified during our CMC development plan to have that meeting. And we I can talk a bit more on subsequent slides or on the specific topics, but we did, get some great guidance from FDA on those topics. Then later last year, we actually had a Type D meeting to follow up on a couple of specific topics that had occurred during the first Type B meeting and kind of expand on further development plans for those. At this point now, we are considering when we would want to have our second Type B meeting under the pilot. And I think given the feedback received from FDA around wanting to see specific further data around our process characterization or process development, we really want to make sure we go into that second meeting with a very clear plan of what we want to achieve, what data we have to support, and where we want to go from there. So that's something that we're currently reflecting on. Next slide, please. So the key development challenges we developed for CDRP engagement fell under the broad buckets of our control strategy, including our starting points for manufacture, our structural elucidation strategy, and our drug product supply for pivotal studies. And our aspiration was, with the increased FDA interactions under CDRP, we can reduce the CMC risk to our program and increase our chances of a seamless and successful launch. So if I move to the next slide, I think one of the things that was specific to our product is really we have a reasonably complex drug substance manufacturing process and supply chain. We've got more than 40 chemical processing steps to our final drug substance. How that happens is we end up with two key intermediates, the bicyclic peptide and the toxin linker molecule that are then conjugated into drug substance in the final step. It was clear to us that we'd want to seek early alignment on where we consider the start of drug substance manufacture, so the regulating starting materials, which are obviously a key topic for small molecules, and to ensure that we had a robust, scalable drug substance manufacturing process. And because of our nature of our molecule, we have different requirements for our CDMOs. So we have, peptide or high potency toxin linkers. And in order to put all this together, you end up with rather a long and complex drug substance manufacturing process. So it was key to us certainly to align on the starting materials and the drug substance manufacturing steps as part of that first CDRP engagement. If you want to move to the next slide, the second thing that was important for us to align on was that the bicycle drug conjugates, once the peptide is constrained onto the scaffold, it's actually remarkably robust and stable molecule. And it can be quite difficult to generate the data you need for structural elucidation looking at the whole molecule. So it's difficult to do amino acid sequencing and mass spectroscopy for those reasons. If you move to the next slide, the approach we took in that case was to really take a modular approach for structural elucidation, look at the linear peptide prior to being constrained on the scaffold and the bicycle itself, and the drug linker, which is more of a standard small molecule and more amenable to fairly standard elucidation. And with all the various analytical techniques that we have used, we are able to build up a picture where we can infer from the sort of the incremental NMR spectroscopy, the full structure of the molecule, and be really confident about that final molecular structure. But it has been something that was reasonably complex and unusual for traditional small molecules that we wanted to get some alignment with the FDA on. Next slide, please. The final piece that we really wanted to discuss was around the clinical development of the product and aligning our CMC strategy with that. So our registrational Duke-Margolis II study in urothelial cancer was initiated in 2023. Aiming for an accelerated clinical development strategy means we had to start that study with the initial formulation used for clinical studies. So still remained a lyophilized drug product administered by IV infusion, but it was stored at -20 in the freezer And we knew we had to redevelop for robustness, stability, and to ensure that we had a suitable product that was scalable for commercial supply. So we wanted to specifically discuss our plans for switching during a registrational study, the product that we would use for that. And I think, as I say, it still remained an IV infusion, and ultimately the changes in the product were relatively minor in terms of excipient changes. The key insight we got from this, and this was actually really useful to us, was to draw our attention to this map 5019.1 around the content and the fill of injectable drug products. This is something that hadn't hit our radar previously. I'll be really transparent, and it's not a guidance for industry; it's a map, so it's typically targeted at the FDA reviewers. And getting that guidance and that insight at that point allowed us to make some very thoughtful decisions in terms of our filling of our drug product and our development of our ultimately what will be our commercial formulation of our drug product. So that has allowed us to do that. So that is a benefit for us. Next slide, please. So as I said, we have had that one meeting under CDRP where we discussed the topics I've just been through. We're now looking ahead to our second meeting and when will be the right point at which to have that. And it's key for us, given the nature of these molecules, that the control strategy is something that's very important for us to be aligned with agencies on. We have, a peptide element, a high potency drug substance, drug linker. We need to make sure that we have something that is scalable and reproducible in terms of control strategy, but also consistently delivers a high quality product to patients. So as we generate data during the drug substance and drug product process characterization, we will generate a data package that will allow us to develop our proposed control strategy and hopefully align with the FDA on that. Next slide. And I think, here just, alludes to those control strategy challenges. For example, peptides are excluded from Q3A and Q3B. We have USB guidance on synthetic peptides. There's an EMA draft guideline that went out for comment on synthetic peptides. We have a number of different parameters we need to consider in our spec and our control strategy. And we have a molecule that falls somewhere in the interface and size between standard small molecules, large antibodies, and it's something that we really want to make sure that we chart a path forward on. And the next slide, and I think it's probably my final slide really at this point. In terms of CDRP, the few aspirations we have for it, and I think, I've heard, great things about that so far this evening. So definitely if there's an opportunity to continue this program, I think it's clear that there is a need for it there, given the number of issues we're seeing with products for CMC issues, and we don't want to, and definitely continue the additional CMC interactions in partnership with FDA. Like to see more opportunities to get input on the CMC development plan and perhaps to consider whether there's more opportunities to participate. For example, CMC complexity isn't limited to products that have an accelerated designation, but they may be related to the modality of the product. And similarly, it may be more experienced sponsors as well. And, we'd like to see just continue to improve communication alignment between the sponsor and FDA. And then I think all that remains to say is, if you want to move to the next slide, thank you very much for your time and your attention today. You can see our facility here in Cambridge in the UK, where I'm based, and to pass back to Thomas. Thank you very much.
Thank you so much, Claire. And yeah, really useful to see, I think, throughout your presentation, not just points where your experience may have been unique, but also I think really useful to bring out some common themes across sponsors who have participated in the program so far. So really helpful on all fronts. Thank you, Claire. Next, we can go to Alyssa Carter for a presentation on behalf of Rayze Bio. Over to you, Alyssa.
Thanks, Tom. I just wanted to start out saying we appreciate being selected to the CDRP program and are grateful for this opportunity today to share the lessons we learned while participating in the CDRP. Next slide, please. Thank you. A little bit about us. Rayze Bio is a Bristol Myers Squibb company. We're a biotech company aiming to become a leader in radiopharmaceutical therapeutics, or RPTs. Within our portfolio of potential RPT candidates, Rayze 101 is our most advanced investigational product in the clinic. And we've had extensive FDA engagement via traditional meetings, as well as the CDRP program, as other people have mentioned previously today. Rayze 101 is an actinium-225 dotatate for injection. It's being investigated for its potential as an alpha-emitting radioactive therapeutic agent. It consists of two active components, if you will. The actinium-225 isotope is one of them, and the dotatate chemical precursor is the other. The drug product is produced in a continuous operation from the drug substance manufacturing through the drug product manufacturing, and the drug substance itself is not isolated. Manufacturing, testing, and release of the drug product must be completed within approximately 24 hours. The drug product is aseptically filled into final patient vials. And the manufacturing process can be performed either manually or using an automated synthesis unit. The drug shelf life is limited due to the isotopic decay. Actinium-225 half-life is 9.92 days. In each dose batch is manufactured for specific patients based upon a physician order. So we have small batches, short shelf life, and a continuous manufacturing process. Currently, there are no approved actinium-225 RPT products, and Rayze 101 is anticipated to be the first alpha-emitting RPT drug product for which a marketing application is to be filed. Our FDA engagement regarding the actinium-225 isotope selection and development started fairly early on in our development process. We attended the September 22nd, 2021 FDA and Nuclear Regulatory Commission, or NRC, joint workshop. It was entitled Enhancing the Development of Targeted Alpha-Emitting Radiopharmaceuticals, and a special session on actinium-225. As a result of the discussions during this meeting, and because of the accepted impurity profile, our early development studies were conducted using thorium-229 generator-produced actinium-225 due to the safety profile and for the safety of our patients. Due to a limited worldwide supply of thorium-229, this generator production method is insufficient to support the growing clinical development across the industry and projected commercial needs of actinium-225-based therapeutics. And as such, additional sources of actinium-225 would need to be identified and discussed with FDA. Thank you. If we want to go to the next slide. I think we already covered that one. Sorry, slide ahead. Oh, sorry, maybe go back. My mistake. As we touched upon today earlier in all these sessions, FDA approval of a new marketing application is based upon data collected during clinical studies, and FDA requires the drug manufacturer to provide data to demonstrate that they consistently manufacture the product with the same characteristics as the investigational product used in the clinical trials. As I touched upon, and touching upon again, a key component of an RPT drug product is the radioisotope. In a restricted supply chain for alpha-emitting isotopes, variability in the methodology used by the suppliers of these isotopes for production and the short half-lives present unique opportunities to pursue with FDA and industry cross-functional collaboration to support meeting the CMC requirements while we're on an accelerated development pathway. As such, additional sources of actinium-225 do need to be identified and discussed with the agency. Next slide, please. Unlike traditional drug products, there are few RPT commercial contract manufacturing facility options available, and technical transfer to an existing commercial facility is a limited option. RPT facilities face additional challenges from the start of the construction to first-in-human dose, and then to support future commercial launch and commercial activities. Some of these are ALERA considerations, which is radiation considerations as low as reasonably achievable. And these need to be built into facility design, and also to consider worker safety. And also NRC licensing of these facilities is required. For the safety of our patients, control of the drug manufacturing process is critical, and we need tight controls on logistics due to the challenges from the short RPT shelf life. And we also need to carefully manage the limited isotope supply. As such, construction of an RPT facility to support phase 3 and commercial product production for our products was needed, and we would need to discuss these with FDA. And these are reasons why we entered into this CDRP program. Next slide. Thank you. Rayze Bio applied in the first round of the CDRP submissions with the goal to have additional engagement and increased communication with the FDA to address our CMC challenges associated with RPT product development. Our accelerated clinical development program for Rayze 101 moved directly from phase 1 to phase 3. And I think this was a common theme throughout a lot of the slides due to the timelines that it took to be accepted into the program. We were already there. We were already moving from phase 1 to phase 3 and starting our phase 3 program. This compressed the time that we needed for CMC development of the commercial product to be introduced into phase 3 trials. So today I'm going to present 2 of the topics for CMC discussion that were held as part of the CDRP and the lessons learned for accelerated product development. The first topic is the isotope supply chain constraints and differences in suppliers' production methods. And the second is commissioning of a new RPT drug product manufacturing facility. Next slide, please. For the isotope supply chain under the CDRP, we discussed the pathways to utilize actinium-225 isotopes from production methods other than the thorium generators that we are currently using in our clinical program and using for drug production. Our outcome from the CDRP engagement was basically due to the limited data on the impurities resulting from the other methods of production, specifically the long-lived isotopic impurities, additional drug product controls, and/or non-clinical and/or clinical studies may be needed before we could introduce the use of actinium-225 isotope from production methods other than the generators. These production methods, such as cyclotron or accelerator-produced materials, are acceptable, but we needed more data before we could bring them into our pivotal trials, as the protocol was already written. So as more data becomes available, Rayze Bio will continue to have discussions through mechanisms like CDRP to address how best to alleviate the restricted supply chain. And these may be production methods that we can introduce later on post-approval for Rayze 101. So our lesson learned was to definitely have cross-functional conversations as early in development as possible through programs like CDRP in order to support an accelerated development program. And CDRP provided this forum to introduce the topic of these alternate production methods for actinium-225. And FDA facilitated additional discussions with our cross-functional teams on this topic. Again, I'll stress that we started early on our phase 1 early on clinical trials with an isotope that had a more favorable impurity profile. So as these new isotope methodologies matured, after we had already started our clinical development program, we were kind of left with finite options due to the acceleration of our clinical program. And so being on the cutting edge and pioneering actinium-225 therapy in the clinic requires real-time modifications to CMC development, and it may also lead to having to make changes to your program that may impact your timelines, your clinical development timelines. And next slide, please. The second topic I want to talk about is commissioning of a drug product manufacturing facility. Again, under the CDRP, we were able to discuss our manufacturing plans to support ongoing clinical trials. And we do have additional future topics such as GMP readiness and commercial process improvements that we'd like to discuss. During our initial interactions with FDA, FDA did suggest a pathway for us to open communications with the local district office to discuss the facility, and also encourage progress updates in future CDRP discussions about where we are with the facility, and to help us with some of the things that were discussed previously about GMP readiness. So CMC does not hold back marketing approval of this application. And again, I stress to industry, everyone listening out there, have your cross-functional conversations as early in development as possible through programs like CDRP to support an accelerated development program. CDRP for us provided this forum to introduce the topic of the manufacturing facility, and FDA facilitated additional discussions with their cross-functional teams. Our plans for future CDRP engagements, we will discuss activities to support the commercial GMP readiness and commercial process improvements for this facility. And we are grateful for this opportunity afforded by the CDRP. Thank you very much. Appreciate it.
All right. Thank you, Alyssa. And I think we can move ahead to the next slide then, and we will jump into our discussion here. And actually, Alyssa and Claire, you can stay on with us, please. And then we'll have two more joining us. That will be Jennifer Swisher joining us again after she provided some opening remarks this morning. As a reminder for the audience, Jennifer is a supervisory pharmaceutical scientist in CDER's Office of Pharmaceutical Quality. And then also rejoining us after participation earlier today is Thomas Oliver, Director of CDER's Division of Product Quality Assessment 1. So both of you can come back on screen with us, please, and we can take the slides down as we jump into the discussion portion here. Starting off, maybe would love to follow up on some of those points that Alyssa touched on toward the end of her presentation around the involvement of cross-functional teams, and maybe get some input from Tom and Jennifer as we start off here on what that looks like from FDA's side, how you all coordinate the involvement of cross-functional teams, and if you want to elaborate at all on the potential benefits of that type of interaction under the CDRP.
Yeah, I can start, and I can ask Jen to contribute as well. So, typically when a meeting comes in, there is someone who basically does a triage to determine the disciplines that are needed to provide effective responses. And typically for many products, it involves on the quality side a number of different disciplines. And we feel that that's very, very important to get that engagement and to bring all parties in, because there are nuances that applicants need to hear from each of the different disciplines during the meeting. I would say on the oncology side, as products are pretty complicated, we have multidisciplinary meetings kind of as I would say a standard. And so we're pretty used to that kind of piece. But again, as you've experienced, doing it early as possible is important. Because I know when you get those recommendations, it's important to have your team kind of think of them and incorporate those recommendations. Jenny, did you have anything as well to add?
No, that's right. I think the combination of the cross-disciplinary team as well as the kind of package, and there's been some discussion of different experiences around the information in the application package, kind of, which gives this broad view, right? So you have the cross-disciplinary team, you have some idea of where the sponsor is in development. And so even if we're still, and we do still require, focus meeting questions, that approach towards answering them can just be that much more interdisciplinary, because a lot of times in meeting packages there'll be questions that we can answer with that caveat that everybody loves, like it's going to be a review issue when we see all the things, because honestly control strategies touch on many different areas. And so when we have that early look at all of those, the different people in the room can really help to kind of, I hope, formulate more comprehensive and helpful advice.
Really helpful. Thank you both. And then let's go back to our two industry participants here. And Alyssa, you touched on the importance of starting this process early in development. And I think Tom mentioned as well how valuable that can be to get that input earlier on in the process. Similar to where we started the discussion in the previous session, can you talk a little bit more about kind of what that preparation work looks like from within your organization, and what steps you all took to ensure that you were prepared early on and to be able to get that advice at an early stage where it can be as impactful as possible. I know you both touched on this a little bit, but if you could elaborate kind of what that looks like working with your colleagues internally.
Sure. At our organization, we prepared for the meeting. We had a thorough review of available resources within Rayze Bio, and we initiated this process when we were still a standalone company with a smaller footprint. And I think there's been a lot of people talking today about the amount of resources and time, and needing to also keep the development timelines going. So it was something that the company had to assess and really back and support for our involvement. We, as a company, understood the importance of these meetings, and we sought to dedicate the right resources to them and expertise in order to best engage with the agency in a thoughtful and productive manner. So, different people, these CDRP meetings were CMC focused. So it was like subject matter experts in RPT, manufacturing, development, chemistry, quality. All of those people were pulled in to prepare briefing books, ask the questions, design the background material so we could have a thoughtful discussion with the agency. And I also wanted to mention in our meeting that we had with FDA regarding the isotope production. So we brought it up as a topic in our initial meeting with FDA, and that meeting topic, after we received the comments back from FDA, we kind of expanded on that, developed some slides, provided that, and the agency did not have adequate time to pull in an interdisciplinary team, because there were clinical components and things outside of this CDRP program. So they really helped us with you know, we can't really discuss this now, but here's a pathway, submit this, resubmit this as a different meeting request, a type C, and provide all this material. And we still cited the CDRP meeting and identify or notified our program manager at CDRP when we submitted the package, so they were alerted to it coming, and seamlessly behind the scenes, FDA pulled together their interdisciplinary team so we could hold that meeting. It was super helpful for us.
That's really excellent. Great to hear, particularly about kind of the handoff and extending beyond the primary scope of the CDRP. Claire, any thoughts along the same lines in terms of your preparation for meetings and even internal coordination with clinical development folks at Bicycle?
Yeah, sure. I mean, kind of aligned to Lisa in lots of ways. We're a small company. We do most of our manufacturing. We do all of our manufacturing through CDMOs. So our core team of CMC experts within the company are very, you know, a tight group of people, and we really partnered together in authoring the CMC development plan, which sort of came out along at the right time for us, because at that point we were really looking at moving from, we'd done our initial clinical studies, phase 1, how we were going to bring this product through development and develop a commercially focused formulation. So this all came together, I think, at the right time as we were making those plans, and we were able to articulate those in the CMC development plan at the same time. So, and that helped us, as I say, clarify our thoughts quite a lot as well. So that was really good. And then, you know, through my clinical regulatory colleagues and through the rest of the Bicycle team, we were able to make sure we were hand in hand with the fast track designation in urothelial cancer and the plans for development of a registrational study in that indication. So we could really draw out what were the key CMC challenges with that as part of that, both the CDRP application, but also as the preparation for that first type B meeting as well.
Thank you very much for elaborating on that. I appreciate the answer. We have a few questions from the audience that I think align closely with some of the questions we prepared, actually, but we're getting a few questions. Oh, go ahead, Jennifer. Yeah.
Questions that did come in from the audience that are kind of related to this. I saw a few that were about like, what other meetings can you have, right? The Federal Register talks about additional CMC interactions. I think Alyssa just kind of mentioned one way that could go. I think for clarity, you know, that, you know, there's opportunities that exist in other types of meetings as well for post-meeting clarification. You know, if there are things that we can't answer in a meeting, you know, there can be emails after, or even after the meeting is ended, to come back with the minutes and say, you know, we have some more questions. But the other meeting types are also not closed to CDRP participants, right? So in addition, you might ask for a focus type D meeting, et cetera, you know, that might kind of flesh things out, and you're probably going to have the same team of reviewers. So I just wanted to kind of clarify some examples of how that goes.
Thank you, Jennifer. I know that's a question that we've certainly heard a lot, so really helpful clarification. And I think Alyssa mentioned that in referring questions to other cross-functional teams, in some cases, it sounded like Rayze Bio flagged that you were participating in the CDRP program and mentioned in those additional requests that your points of contact in the CDRP had directed you to send this question along. Is that Tom or Jennifer, would you want to confirm if that's kind of best practice for CDRP participants to try and facilitate some of that coordination from their side as well?
Yeah, I think it's always important to do that. I mean, to have, you know, things that you can do, you know, if you're doing, as Jenny said, you're taking advantages of not only the CDRP program, but you're taking advantage of, you know, some of the other meeting types that we have, like Ds. You know, it is important to try to keep on your side the same team involved. So if you knew that a team was previously working on it and including that and recommending people to be part of it for continuation, that's something you can do, as opposed to, you know, inadvertently getting another team assigned to it. We try on our end not to have that. We try to keep the same team involved through the process development, but that's something that we see sponsors do to try to prevent that happening. But anything you can do on your part to notify us, the people that have been working with it previously, it's advantageous to do that.
Makes sense.
If I may, I might just add to that a bit with our experience as well. So we had, a successful Type B meeting in early 24. We had a couple of follow-up topics and how we, just from purely from mechanics perspective, utilized the follow-up with FDA. We went back to the same project manager on the CMC side and said, look, we have these topics. We're in CDRP. We think it's a Type D. Do you guys agree? And they were able to say, yeah, that was an appropriate way to get clarification at those points. So that was useful for us. We're with the same team on our side, but also, if I recall correctly, it was the same reviewer on that Type D meeting minutes that had been involved in our Type B as well. So that was, something that was obviously advantageous for everybody.
Fantastic. Yeah, that's great to hear. Thank you for adding that on. A couple of questions from the audience, maybe a little bit more specific to some of the products we're talking about here. We did get a question that I think was directed toward you, Claire, regarding regulatory starting materials that you touched on a little bit in your presentation. An audience member asked how the strategy and validation of GMP steps might have looked different in the case of the peptide as opposed to, I think, comparing to other products there. And they also asked whether there was any flexibility offered due to participation in the CDRP. Anything more you can share on either of those points?
Yeah, so I guess, with a relatively complex molecule, we needed to think kind of be very thoughtful about our selection of regulatory starting materials. For the peptide element, there is our existing guidance, for example, in USP 1503, that talks about what you should consider as starting materials for synthetic peptides therapeutic. So we were able to, utilize that guidance, look at our other intermediate, our toxin linker, and come up with a proposal. I guess the key point to say is at this stage of development, that's really a tentative assignment because it's pending completion of all your process characterization data in line with, ICH Q11. So that is kind of an ongoing piece of work for us, but we got the feedback we needed to be relatively comfortable that we're heading in the right direction, pending completion of all that additional data generation as well.
Thank you for clarifying there. And Tom and Jennifer, would you want to comment any further on kind of in general how FDA considers any flexibilities that could be offered in the development process? I know this was something that was touched on a couple examples in your presentation earlier, but we were getting quite a few audience questions for further clarification there, without getting into specific examples product by product, but how FDA thinks about offering flexibility, particularly when working with sponsors who are in the CDRP.
Sure. You want to go first, Tom?
You can go first, Jenny.
Yeah. All right. So, sure. So I think the most important thing, and I think I tried to really emphasize this, right? Because we get a lot of requests that start with, we're really early, things just got expedited. We don't have much information. We need a lot of flexibility, right? And so that doesn't work out too well, right? There needs to be the product and process understanding, even if it comes from prior knowledge, even if it comes from, early phase manufacturing, that type of thing, to kind of begin to inform those discussions. So I think in thinking about a control strategy, I do think for CDER products, that OPQ internal map, this is our policies and procedures, but it's a way, we've published it externally to let folks know this is our thinking. Lists a lot of commonly used or commonly proposed flexibilities and talks about kind of the basis for approaching those, right? In terms of validation approaches, stability approaches, different things. But it really, again, it's really predicated on kind of product understanding, that you're picking a manufacturing site that's, fully GMP compliant. There's no flexibility there, right? So, and that there might need to be kind of extra testing, extra control other places. So that's just kind of a general approach. There have been a lot of specific questions, and there really are a lot of specific answers in that map. So I would direct people there as a starting place. But if you think you've got something else that's justified in terms of the science and the justification that you can make and the data that you bring in, that you can, satisfy the regulations with a program that's going to have some flexibilities in some places, but safeguards in others, to make sure that you can demonstrate that that's going to work to assure consistent quality of the product. That's just the best approach.
Fantastic. Thank you.
Tom,
Tom,
anything to
anything you
add?
wanted to add?
Yeah, I would just add to that, adding kind of to what Jenny said, we still need a control strategy, and we still need, mitigation of gaps. It's not like, a product that is, in the breakthrough space basically doesn't have certain controls in place. It doesn't give in tremendous flexibility without mitigation. That we have we might not ask for traditional data elements. We may be willing to work with you on a patchwork, but we still have to have confidence in the control strategy. We still have to be confident in the expiration date. We don't want the product recalled off the market because patients are exposed to some undue risk. We want to make sure we have confidence in the product and how it's controlled. And so we often in this space are a little bit more flexible, taking data that you've collected, maybe not the standard data that you would normally submit for a BLA or an NDA. And so that's why it's so important to engage the agency with these types of products to make sure that you understand the types of data we're willing to accept and work with you on additional data that you may not have that you would be able to generate in the time that you're collecting information on your product. Thank you.
Thank you, Tom. And then turning back to our industry participants here, I just want to revisit some product-specific questions here, because there really is, I think, so much that lessons that we can learn from what sounded like pretty demanding CMC development processes in both of your cases. I'm particularly thinking about the fact that we're talking about some products with shorter shelf lives, manufacturing relatively smaller batches. In both cases, you talked about some complexities with starting materials, inputs, finding suppliers for these all factors that kind of add additional compression to the timeline and the importance of timing on various stages of CMC development here. Can you talk about some more of those particular to each of your products and how you work through things like the shipping logistics, finding and coordinating with manufacturing sites, anything along these lines?
I'm happy to start with a few initial thoughts. I don't, I think for our BDC molecules, we don't have a particular shelf life challenge. They're actually, relatively stable molecules, but what they do have is a complex drug substance supply chain with different CDMOs in different locations. So we need to be able to make sure that we can coordinate our supply strategy through those, but also that we have, the right sort of oversight. And given the length and complexity of our manufacturing process, that's something that's important for us. Important for us to ensure, and it's all about that kind of mentality of building the quality design built in end to end, through the manufacturing process.
Thank you, Claire. Alyssa, anything to add?
Yeah, it was shipping logistics. I mean, part of why we went to commission our own manufacturing facility is to have more control over that type of process. We're the ones responsible for the actinium-225 coming from our suppliers, and we want to ensure, because of shortage, ensure we don't run into a shortage situation that's tightly controlled, and it just helps us to manage a lot of those pieces by having it under our own internal controls. There are some common themes that occur with RPTs, and FDA has talked about these in a lot of different industry meetings and forums. Things about shipping under quarantine, conditional release due to the short shelf life, and also because of the fact that the product is released before sterility results are available. Just discussing with them designs of media fills, establishing appropriate bacterial and endotoxin limits, things like that. these are all questions that industry as a whole, can go through a mechanism like CDRP to help get some valuable feedback early on in their development program. That would be my advice to industry.
Mm-hmm. Well, and building on that point, I think in both of your cases and some of the other examples we've heard today, we're talking about more complex products or modalities, and in some cases novel products or modalities. And we got a question from the audience about this as to kind of whether that is the exclusive focus of the CDRP program, whether that's the only types of products where we could see a benefit. It sounded like, Alyssa, you were saying there are certainly some benefits here that could apply to plenty of sponsors across different types of products. But before we run out of time for this session, I wanted to pass that general question back to our FDA participants. Would products that are less complex than some of the ones that we've discussed here, for example, monoclonal antibodies, maybe with a better understood mechanism of action and quality attributes, also be eligible for the CDRP? And can you talk a little bit about, if so, what sort of benefits those sponsors might see?
Sure. Yeah, no, complexity is not a requirement, right? In terms of, it certainly does present challenges, right, that are well suited to kind of dealing with by this program. But really the overall challenge is the acceleration of the clinical program. And in kind of our analysis of what they're going through and what the particular needs are, a monoclonal antibody could absolutely qualify if there were particular challenges towards something that was really clinically expedited and prioritized to meet whatever those particular needs are. That hasn't typically been the case in the applications, right? There are a lot of things published, and there are a lot of kind of approaches that are pretty standard. There's a lot of prior knowledge for monoclonal antibodies, and so it's less likely, but it absolutely could be a good candidate.
Excellent. Thank you, Jennifer. Tom, any concluding thoughts there with our very final minute? Anything further on that point?
No, I would just say from what we've seen, and it's a good example from what Claire and Alyssa brought up, each product has its own challenges. And those challenges, through the CDRP program, you're able to kind of get at what the issues you have to deal with in a kind of a direct way early. And I think it's very beneficial. It's kind of a unique CMC only program that we have. It's not like kind of a general program that currently exists for breakthrough or some of the other accelerator programs. And so it's a way for individuals with a unique product and unique challenges to work with the agency on those challenges early and develop like mitigations or strategies going forward with the ultimate submission of the application.
Thank you, Tom. A great note to end on, I think, and a good segue to our next session. Thank you again to our presenters and all of our panelists here. A really great discussion. And now I will introduce my colleague, Brian Cantor, to come back on with us, and he will be moderating our final panel discussion here, building on some of those same points about how the CDRP can continue in an impactful way to help sponsors overcome CMC challenges. Brian, back to you to take it away.
Yeah, thank you, Thomas, and thank you to all the participants we've heard from today, and thank you to the audience that's stuck with us into hour 4 of the event. And we've had a great discussion so far. We've heard a lot about how the accelerated clinical development timeline really requires adjustments from the CDC development, and sometimes that accelerated clinical development timeline is broadcast widely and made aware to the CMC team, and sometimes there are some information gaps, but either way, there's a lot of uncertainty that comes with CMC development, especially for some of these complex products. And the CDRP program can really help with de-risking on topics related to comparability and stability. And FDA has also benefited really from being able to have conversations with sponsors through the CDR program earlier in the development process, and being able to have conversations to mitigate some of these CMC issues earlier. And this early engagement really helps towards defining flexibility, especially from a data standpoint, and ensures kind of transparent communication between FDA and the sponsors. And it also requires the sponsors to work with their other manufacturing partners, whether that's CMOs, when relevant. And we really heard in both of the prior sessions about the benefits of informal feedback from FDA to sponsors, both within kind of the CMC related and Type B meetings, as well as in other venues. And we've also heard about how important it can be for briefing documents to include things like focused questions to help organize some of these discussions. And also that there's been a number of resources that are and people and time that go into preparing for the CDRP program. And there's still additional steps that can be taken to clarify some of the value for the CDRP program in the context of some of the other expedited review pathways. And so in this final session, we'll reflect on conversations we've heard so far and continue to discuss CDC development more broadly and some potential next steps for the CDRP program. We'll continue some of the dialogue between program participants and FDA around facilitating CMC development of products charged for expedited regulatory pathways. At this time, I'd like to invite our panelists on screen as I introduce them or reintroduce them back to the audience. We'll welcome back Claire McCarthy from Bicycle Therapeutics. Hope, Claire, you didn't go too far. And Isabella Palazzolo as well from Intellia Therapeutics. And then in addition, we'll be joined by Celeste Frankenfeld-Lamb, who is the Senior Director at Merck & Company, as well as the co-lead of the International Society for Pharmaceutical Engineering Product Quality Lifecycle Implementation Team, who she's representing on this panel. We'll also hear from Cecilia Tem, who's the head of US CMC Regulatory Policy at Genentech. And then our final panelist is Stephen O, who's the Acting Director of CBER's Office of Cellular Therapy and Human Tissue, to give the agency's perspective. And I will get started with a few questions, and a reminder to the audience to continue populating the Zoom Q&A function with questions throughout the session, and we'll get to as many of them as we can. So I think I want to start with to kind of each of our panelists, what were some of the key takeaways you heard from today, and where do we see some opportunities for kind of learning or improvements to the CDRP program? And I want to bring in Celeste maybe first, as you've kind of can reflect on some of the discussions from earlier sessions.
Absolutely. So, and Thomas was just talking about this, but I think one of the things we heard over and over again was the value of the informal discussion, that informal engagement back and forth. Even just today, listening to some of the answers to the questions that we received. You know, I took a note here from Jen where she was talking about that there needs to be product and process understanding, even if it is, you know, based on prior knowledge. I think when we hear these off-the-cuff remarks, it provides additional context and color. And that's what's really meaningful to manufacturers as they are trying to develop these processes and get them to the patient so quickly. Clarity, right? It's the informal discussions and it's the clarity that are so valuable.
Thank you, Celeste. Cecilia.
Well, thank you. So to me, I really appreciate this workshop because I think that provided what you were saying, Celeste, clarity about a lot of things that sometimes are not that clear from the information that is out there. The fact that all the additional or the regular meetings are available as well, and the restriction is not only on 2 Type B meetings, and then you can really leverage shorter timelines or more focused discussions like using a Type D meeting, that really shrinking that time from where you have the question and where it is addressed is going to be very helpful. And these informal interactions as well. Sometimes, I mean, time is very precious, and so having kind of a feedback that is sometimes it's even minimal, but has a huge impact in decisions that are taken on how to move the program forward are key. So again, this frequent potential informal interactions and partnership that I think I heard through this workshop. You're
I'm
muted.
muted. Had to be the first one to get that done. Checked off the bingo board. Stephen, I want to bring you in as well to see what really resonated with you, and maybe something that you expected to hear but maybe didn't.
Yeah, sure. So in my case, what I heard from each case study examples, especially, is that the science and risk-based approach was the key driver for facilitating the engagement with the FDA early in the development, or as early as possible in the development. Then the driver that allowed these types of focused discussion seems to be the well thought out CMC development program that was in place with the package. And that development plan obviously was thinking towards commercialization of the product as early as possible again. And because of this early understanding that FDA has in terms of the overall CMC plan and the comprehensiveness of that plan, it seemed like FDA was able to provide insightful comments and directions, but also was able to use and think about some of the options that could be considered as regulatory flexibility, some of the tools and options that could be possible for that particular development program. So I think those are some of the key takeaways that I heard from the case studies.
Yeah, thank you, Stephen. And you referenced the the comprehensiveness of the CMC development plans prepared by the program participants, and got a really great question here from the audience about how what sort of the potential is for participation in the CDRP program on the sponsor side to help uncover unknown unknowns in terms of CMC development challenges that would have to be overcome to have an approved marketing application, and considering issues that may only arise during the development process. Claire, Isabella, any thoughts?
Yeah, I'm happy to start. Maybe Isabella, you can chip in your thoughts as well. I think that's exactly what we were hoping to get out of the program. We had working with an experienced internal team at Bicycle who've done developed products, commercial products with different organizations, but we really wanted to see what were we missing, what could potentially trip us up, because the last thing you want is a terrible surprise at a crucial part of an NDA review, which could be catastrophic for a small company. The guidance we got, so I think we were pleased that we saw and we were able to have discussion on some of the CMC topics that we had pre-identified as being something we'd want to discuss, and that was really useful. And then I think, as I alluded in one of my slides, during the discussion on the development of our commercial drug product formulation, we were given a heads up from the FDA about that additional map around the fill volume and content. And when you think about your filling of your drug product, that's quite hard to fix at the time of an NDA. You've already manufactured batches. You end up, situation where you have to change your label strength, or it's a really challenging issue to address. So for that was the key unknown that came up for us. That was very useful in those discussions.
Yeah, thanks for re-upping that example, Claire. Isabella.
Yeah, I think from our end, I second everything that has been said. I think I want to echo also the idea of being science and risk-based approaches. I think some maybe a separate topic from the unknown unknowns, but as part of our discussion, we really kind of put a lot of these risk-based approaches in our briefing books, and we were able to engage in a conversation that spanned throughout multiple of our interactions with FDA on common positions, on the same topic that we kind of brought back a couple of times to really reassure that, like, did we cover all the unknowns? So those are kind of like the field, those are the topics that we know the least amount, and like, are we on the same track? Are we on a good track, on the same path, and on the same understanding? I think it was like among all the recommendations that the FDA provided us for topic to consider, that like kind of like came as a little bit of an unknowns was like the level of details to be provided of the lipid excipients, especially the non-compendial lipid excipients, and that has really like it was good timing to have that discussion, so we could set up like work in motion to make sure that we have the right approach comes BLA time, because those are like I really like, those are the things you don't want to be surprised upon.
Yeah, thanks for sharing the benefits again of that transparent communication, Isabella and Cecilia.
I just so the thing that to me was very interesting was, I think Andrew mentioned that having this understanding of the end-to-end development plan and being able to identify those places where more input could be given by the FDA, more guidelines, so that you kind of try to minimize risk, increase predictability, and then reach to a BLA that is successful, or an NDA. So this is something that I didn't appreciate, but it came as a very good benefit, and in why all this development plan that I think, Isabella, you mentioned, is quite challenging and resource demanding, is really helping throughout the development, hopefully.
Yeah, no, thank you for mentioning, Cecilia, the end-to-end development perspective that could be seen through the CDRP program, and wondering as some of these unknowns become more and better known, and the CDR program continues to evolve, and Stephen, thinking about how the FDA is able to collect information from the experiences with a number of different pilot participants. How can the CDRP continue to evolve to more effectively continue to meet the needs of industry sponsors? Yeah, I think the second part of that is sort of on the sponsor side. What are some of the more pressing challenges that would expedite CMC development? But Stephen, maybe you can start.
Sure. So do you mind repeating the question again? I think I got that, but I wanted to make sure that I'm answering it the right way.
Yeah, just maybe you can comment a little bit on kind of the evolution of the CDRP program, as there's kind of more known information on unknowns, on previously unknown factors that from past CMC development programs.
Yeah, so that's so I think that's where the beauty of CDRP program lies. In a lot of the traditional CMC discussions that we have through the other types of meetings, the conversation and the briefing package typically centers around what the sponsor thinks things are missing or what could be challenging, and the questions and the briefing package starts there. Now, the CDRP program, as I mentioned earlier, with the more comprehensive development program submitted in the package, FDA has ways to look at these types of rather comprehensive program and the timeline, and be able to really see what might be a blind side for the sponsors, and those are kind of unknown to the sponsors, whereas it might not be necessarily unknown to the FDA. So I think the benefit that you would see, this sort of benefit that you would see in CDRP, is something that you would probably not see in other types of meeting. programs that we have.
Yeah, thank you for that, Stephen. And maybe another thing to think about for the broader panel is how can learnings from CDRP participants be disseminated to improve readiness kind of across the broader industry, for complex modalities, but also, as we touched on kind of the last panel, also for modalities that have a little bit more well-defined standardization when it comes to manufacturing. Celeste, I don't know if you have any thoughts here.
I do. And I think it also ties to what Stephen was just speaking about, as we're thinking about what the FDA is learning. I think examples will be really helpful. Obviously, this workshop is a fantastic start at that. I want to share my gratitude to those of you who have come and shared your experience, because it helps inform other companies who are looking to accelerate, and they can see your experience with this. And I think whether the examples are published by the FDA, and perhaps these are theoretical, maybe you combine real world examples to share, if you have this type of information and you are facing this type of challenge, this is how this flexibility may be applied. Or, the examples could also come from each of these individual companies where they're able to publish, their experience. That provides a map for other companies, and puts it out there in the public arena so that people can learn. I also think that it's going to be important for FDA in their global leadership as well to share how they've been approaching the CDRP program with other regulators that you engage with. There was a brief discussion earlier today about harmonization, and I know that there are many regulators who are interested in enhancing accelerated programs within their own countries, but they don't quite know how to do that. And I think FDA can serve as a model leader there as well.
Yeah, thank you, Celeste, for bringing up again the international harmonization and piece, and highlighting the importance of FDA in terms of global leadership. I think another place to maybe dive into too is looking back again at Claire and Isabella, thinking about your participation in the program. What if any of the recommendations from FDA were surprising, and in terms of how they may have caused some adjustments and extensions of the timelines that were longer than you all had previously planned?
I mean, I don't think that was a specific experience for us at all, to be honest with you.
Yeah, I would concur. I think there was a topic that wasn't planned that we didn't have on our radar, but it did not delay our timeline. We had time to address, and then we were grateful for the agency to bring it up at the right time.
Yeah, thanks, Claire and Isabella. And Isabella, your audio, at least on my end, was a little staticky, but I think still got your message coming through. And I think something else that kind of related to sort of the timeline piece would also be how can, from the sponsor side, how can there be proactive planning to generate the comprehensive needed CMC information on parallel timelines, and included in relevant regulatory submissions. And that could be both for being accepted as part of the CDRP program, but maybe also there's some interest in how some of these CMC development and readiness can be improved for sponsors that either aren't accepted or choose not to apply to the CDRP program. And I know I didn't address the question to anyone specifically, but open to any panelists.
Was the question around if even for sponsors who are not on CDRP, how they could sort of utilize some of the advantage, some of the things we've discussed today?
Yes.
I think it's CDRP is brilliant, and I'm so thrilled to have been part of it to date, because actually there's very little that is directly focused on CMC. You have a lot of excitement about accelerated clinical timelines, and then you see data showing actually 3/4 of CRLs are due to CMC. So something that's addressing that disconnect is really exciting. I think, probably the lessons for other sponsors here today are not a surprise to anyone. It's just getting the internal alignment and the strategic planning together around planning early. I would say even the concept of writing a CMC development plan was helpful for us, because it structured our thinking and our thoughts around CMC development. So having some sort of a roadmap for your CMC development that you articulate within your organization to allow you to plan for when you would want to have agency interactions might be something that people may want to consider.
Yeah, thank you, Claire. And then to maybe just shift topics, as I talked a little bit about CDRP, evolving to meet the continued needs of sponsors, but thinking about, as kind of new technologies and tools come online, and thinking specifically maybe about AI and machine learning enabled drug development, and thinking about model-informed drug development or digital twins for manufacturing, kind of what considerations would FDA giving to those tools being used, or also what could sponsors that are prospective applicants for the CDRP, what considerations are they giving to those when they're as they're planning their CMC development. But Stephen, I don't know if you have any thoughts on kind of from the FDA perspective on industry use of kind of the AI or ML enabled drug development tools.
Yeah, sure. So FDA reviews technologies that use AI ML models, particularly to analyze in the submissions the sponsors use to analyze manufacturing data, controls, and testing data. So we review these types of technologies and data, just like other CMC information in the submission. So this would be something that would really be part of the CMC readiness, development readiness program as far as FDA is concerned. So if any sponsors trying to use AI ML model in their data analysis or any types of control, I think it is in their best interest to really talk about this with the FDA as early as possible. Some of the examples I can think of for using AI machine learning models, at least for the cell and gene therapy products that I'm more familiar with in the FDA, that you will see them in for example, patients' NGS data analysis, which is used to design a product, patient-specific product, and that information coming out from the use of AI ML is used for not only for manufacturing control, but also product design, as I just mentioned. So those kind of uses are real, and there are publications about this, and there are people trying to develop this for product development. And I would also point to FDA's latest guidance, draft guidance document. I think it was published this year. Earlier this year, and the title is something like the use of AI to support regulatory decision making, and these are applicable for drugs and biologics. So though that guidance document talks about credibility assessment framework, and it is a risk-based credibility assessment framework for AI ML models.
Yeah, thank you, Stephen, for bringing up the cell and gene therapy example and referencing the recent FDA guidance. I didn't know if there was any perspective on that from some of the other panelists on, you know, how AI or ML enabled drug development tools may fit into kind of, you know, CMC development and readiness in the context of applying to the CDRP program.
I think there are some opportunities there simply for, you know, there have been a few references made to the FDA map. I think it's, I'm not going to name the number because I will probably get it wrong without looking precisely at my notes, but the map on CMC readiness. And one of the topics that's listed within there is predictive stability, or you know, that is a form of modeling that we can use to, with the appropriate knowledge about your product. Maybe it's first principles, maybe it is, you know, a model you've built off of the development stability that you have throughout the course of that product. But that's obviously one of the ways that you can use machine learning or modeling for supporting CMC acceleration. I think there are other more sophisticated uses as well. As you were trying to model the manufacturing process, I think you referenced digital twins earlier. You know, those have the potential to, again, accelerate the development. You can collect data if you have a robust model without actually running some of the experiments. So I certainly think that there are opportunities for AI and machine learning as you look to accelerate the CMC readiness. And there are other opportunities to engage with the FDA as well. There's the CDRP, and I think there are other, you know, emerging technology team or the CAT in CBER to also have those discussions, which builds upon some of the comments that others have made earlier today already, that, you know, you want to pursue that multi-pronged approach for that engagement with the FDA as you're looking to move forward.
Yeah. I didn't know if you had anything to add.
So I think that really in the context of the CDRP, having the opportunity to work together with and get a lot of feedback from the FDA is very helpful when you have something that is novel, a novel modality, or a novel approach, like you were mentioning, Celeste, about predictive stability, or something that really you don't have a lot of experience with, and maybe there is not a lot of experience necessarily out there on how to use it in regulatory context. So I didn't think about AI and CDRP necessarily before this question, and I really appreciate it that you bring it up. But I do think that novel molecules, novel approaches is really give a really good opportunity to be into the CDRP pilot, and then get more feedback from the FDA and more discussions on how to move this forward.
Yeah, no, absolutely. And appreciate that, Cecilia and Stephen and Celeste, you kind of both mentioned how, you know, involving topics like AI gets the need for expertise and other you know, resources outside of just the CMC space, and had a discussion in previous sessions about the importance of cross-functional teams, and maybe can start here with Claire and Isabella about what it sort of took maybe within your own kind of organizations to be able to champion bringing together you know, cross-functional teams from you know, different divisions and different expertises, and know that's not always something that's very easy.
Okay. I can try again. I can start this one and try if the audio is better.
It's good. It's good.
Yeah, it didn't do anything. But So I think from I guess from our side, what I really made it easy for the regulatory team at Intellia to consider the application to the CDRP program is having a very robust CMC development program already in place. We had a very good plan, and we've always been executed with the 2-year window, the 3-year window, the 5-year window. So we had already some of the material already available. I think what was maybe more challenging, I want to say, is maybe a shift in mindset between having that information available to guide your internal development and put it in a briefing book that goes to the FDA. That was probably maybe more of a shift in terms of like, are we doing the right thing, or are we? This was getting us out of the comfort zone, and I'm very glad to hear that actually it panned out, and like it really did provide the FDA that overview that otherwise they wouldn't get, and provide the FDA an opportunity to also influence where we would need it to go to stay on track.
Yeah, thank you for that, Isabella. Claire.
Yeah, I think from my perspective, it sort of happened exactly the right time for us. We had just started to build our internal CMC expertise, looking forward to commercial development. And we had, a nice team of subject matter experts within the company. And we were really looking forward, as a small company based in the UK, to what we need to do to enable a submission to the FDA. So for us, it was actually, I guess, a bit of a no-brainer that this is something we would be interested in. And it was just a matter, I think Isabella's right. I think there is a bit, it's quite uncomfortable to write a submission to a regulatory authority going, we might do this, but also we might do something else, and it depends what the data will tell us, because that's not normally how you do that. It's normally writing in past tense. So about your development activities. So yeah, that was a little bit of something we had to get our heads around. But I think, overall, people were on board, and it was worth it for us.
Yeah, thanks for that reflection, Claire. And want to jump back, if we can, a little bit to the international harmonization piece, which came up earlier in this discussion and in other sessions, and maybe would be helpful as we get towards the last few minutes of this session to think about kind of what are the opportunities for FDA and industry to work with other regulators just to ensure that longer timelines in one region don't impact or cause issues and availability in sort of other regions, and acknowledge some of the ongoing efforts that were mentioned earlier through organizations like ICH. But Stephen, maybe you could start.
So I think Andrew Burns mentioned this earlier in one of the earlier sessions too. I think that FDA has several mechanisms that we interact with our international counterparts. We haven't really had a specific discussion about CDRP program itself, because this is a pilot for us, and we're only in the midpoint of this pilot program. But there are definitely interests that surrounds the topics that would be surrounding the CMC readiness for marketing application, and how that would look like in terms of product development. So we have ongoing discussions of this type in various venues, some more formal, some very informal. So I just like to leave it at that.
Yeah, thank you, Stephen. Celeste, didn't know if you had any thoughts here as well.
Well, I think there are opportunities in some of the international forums that FDA is already participating in. ICH, ICMRA is also a prime meeting of the minds of the regulators. And then also some of the cluster meetings that the FDAs have with other regulators. That's an opportunity maybe to discuss some of these programs that are being submitted to multiple agencies to share the acceleration. There's also some collaboration between FDA and EMA. The EMA has their PRIME toolbox, where they have a lot of highly similar flexibilities that they offer compared to those that are listed in the FDA. And there might be an opportunity again for FDA and EMA together to provide some of that knowledge that they've built through the use of these flexibilities to some of the agencies that are growing and maturing. And again, ICMA may be a place to do that.
Yeah, thanks for overviewing those venues, Celeste. And then another good question from the audience. It might be a good spot to end on. And I think Claire, you referenced kind of earlier the interactions or with the CDMO that were used by Bicycle, but knowing that the CDRP program is really designed to help smaller sponsors that may be using CDMOs. Maybe you can speak to a little bit of some of the engagement with the involvement of the CDMOs and your engagement with FDA.
So probably not something that we specifically did, I think, as part of our CDMO process. Obviously, we work very closely with the CDMOs when we're as part of our process development and process characterization and working with them to, but in terms of involving them in the FDA interactions or the CDRP piece, it wasn't something that we did. Now, I would say that we didn't really get into the facility readiness piece at all. And obviously there's a key piece around, where there are challenges with contract manufacturing organizations being a reason, and PAIs being a reason for CMC issues with NDAs and BLAs, but it's not something that we specifically engaged in, really.
Thanks for the clarification. Well, I think we are about at time for this session. I want to thank all the panelists here for your perspectives and comments and throughout this session. And now I'm going to turn it to Mahesh Ramanathan, the Deputy Director for the Office of Policy for Pharmaceutical Quality within FDA's Center for Drug Evaluation and Research, to close out our fantastic half day of discussions. Mahesh, please go ahead whenever you're ready.
Thank you, Brian. And what a great discussion we've had here today. So I have the honor of providing just a couple of closing remarks, and really starting with a lot of thank yous. So sincere thanks to our colleagues at Duke-Margolis for hosting today's session. We couldn't have put this on and had such a robust discussion without them. Also sincere thanks to the industry panelists and their FDA staff for preparing for this discussion. A lot of work behind the scenes to really ensure that the content presented really reflected the learnings that we've had for CDRP along the way. And also to all the participants in the CDRP program pilot to date over our first 3 years, who are brave enough and enthusiastic enough to engage with FDA and putting their name in the ring to put their products forward. So we've certainly come a long way from the beginning of PDUFA 7 and have a lot of good experiences in these first 3 years of implementation. And I'm reflecting on some of the conversations we had during PDUFA 7 negotiations to really understand some tools that we could put together to address these concerns around accelerated clinical development and the impact that it has on CMC development. And really envisioning CDRP as a way to create a forum for FDA and industry to engage on these specific hurdles with CMC development acceleration, the strategies to overcome them, and also ensure that the applications, when they came to fruition and came to the stage of filing, were complete and all the regulatory requirements to have a substantive application that can be reviewed and ultimately fileable with the goal of providing accelerated review and accelerated access to therapies. Throughout today's discussions, we talked about what we learned over the first 3 years of this program. We've heard the themes of finding flexibilities and meeting the data requirements of submission, elements like manufacturing facilities, CGMP readiness, how things might move across different facilities, as well as recommendations for applicants on how to best present the need for such flexibilities throughout the development program. And we've also heard that while there was effort required to prepare for and strategize these engagements, while also planning out the CMC trajectory and the development, CMC development trajectory, that the benefits of alignment and FDA feedback were worth it to ultimately reduce some of that CMC uncertainty, development uncertainty. These experiences demonstrate the value of early engagement and timely alignment in reducing CMC development uncertainty despite an overall accelerated timeline, facilitating readiness of submission, and ultimately ensuring that there can be collective confidence in the quality of drugs distributed to patients. So we have some next steps ahead of us. So we want to react to the discussion today and react to any comments that we received in the docket supporting today's meeting. And all this, including the experiences that we've had over the past 3 years, will inform the forthcoming strategy document that's also part of this PDUFA 7 CMC commitment. The goal of that document will outline the agency's plans for to develop or revise relevant maps, recipes, and other applicable documents, for example, guidance or process documents, to incorporate lessons that we've learned across the pilot implementation. So a lot of work ahead of us to really benefit from the good knowledge and good learning that we've gained over this pilot implementation. So let's continue to work together to enhance and facilitate CMC development under accelerated timelines and ultimately ensure that patients have access to reliable therapies for clinically significant needs. Thank you again for all your time and attendance here today. And I'll bring it back to Duke to close this out.
Thank you so much for those words, kind words, Mahesh. I want to echo your thanks to all the participants and also the emphasizing that it's going to take a shared collective effort towards ensuring that patients have access to those needed medicines. Want to get a little specific about, thanking FDA staff who played an instrumental role in planning this meeting. Many thanks to Sunny Kim and Ram J. Watson, Thomas Oliver, Tanya Clayton, and Danielle Villada. I'd also like to thank Duke-Margolis team, including Cameron Joyce, Thomas Rhodes, Matt D'Ambrosio, Garrett Hamre, Molly Shields, Maddie Cordell, Luke DeRocher, and Hannah Vitello for all their efforts to put this event together. As a reminder, additional meeting materials will be posted to Duke-Margolis website in the coming days, including a recording of this workshop. Thanks again for a wonderful event and have a great afternoon.